go from here. Can you guys see a green alien?
Yes.
Excellent.
Yes.
Thank you. So Robert gave you my background. What I didn't put in the bio was that I'm a little bit of a freak. I like to do the hokey pokey in a boot bath. My password's been loved and necropsy. I really love what I do. So I'm just going to start out letting you know I'm a bit of a freak on some of this. The other thing I'll let you know is that my husband has a kidney transplant. And so a lot of this comes really home personally to me. If any of you have someone in your family that's pregnant or undergoing chemotherapy, immunosuppressants for an organ transplant, you know, those of us who are in those high risk categories, and I mean, the terms high risk right now are certainly common. When I have historically given this training, we talk about masks and exposure and positivity rates. So the fantastic thing is that you guys, just in the popular press right now, this is on everybody's mind right now. So it's a really good time to think about it in terms of our work, and in terms of what we do in our regular non kind of shelter in place or telework status, and to think about it, because a lot of risk out there, and if we make smart choices, we can prevent disease spread amongst our wildlife populations and prevent disease spread to people as well. So an old famous dude made a comment about wildlife disease control. It's a matter of doctoring the environment and not doctoring the animal. So I'm going to ask you guys, again, feel free to pop off. What looks funny here? Anybody see what looks funny?
Pelican's laying over on one side.
Yeah, we have a tipsy pelican. What we really need to be thinking about is taking a step back and looking at the larger landscape, all the different potential drivers. So what do you guys see in this picture?
A lot of cows.
Yes, we've got domestic animals there. We've got a lot of cows right there. Good point. We've got a mix of wildlife and domestics. Great point. Other things.
Maybe some water that doesn't have a lot of fresh flow into it, so it might be a little stagnant.
Absolutely, good point. So if you and certainly we don't know what the inflow and outflow exactly are, but looking at that landscape, it looks kind of like it might be a closed basin or a pothole type of system. Excellent observation.
Are those babies? I can't tell.
So there are different ages of birds, for sure, both different ages and species. In fact, there's a breeding area right near there. Good points, guys. Really good observation. So one of the things to think about too is the surrounding land. From what we can see in the front, it looks mostly like grassland. So there's probably not a huge load of pesticides or fertilizers being used. I know we can't make that assumption, but we kind of look around in the background and there's probably some more intensive ag fields in the background as well. So we could presume that there might be some pesticide and or fertilizer runoff into that potentially closed basin of water. So excellent job, you guys, really. Most people are like, I see birds and cows. Excellent. We talk about some of the basis of epidemiology, disease. We talk about the epidemiologic triad. This is something that's kind of in those classic disease classes. This probably got invented in the 50s. I haven't dug up who invented this concept, but it makes sense that you have this disease agent, or a pathogen, sometimes we call it, and the host, which is the animal or person that the pathogen affects. And then the environment, and the environment really plays a huge role. And in fact, if you think about it in reality, we kind of flip this over because the relationship between the agent and the often teeters depending on the health of the environment. So in healthy populations, they tend to be more resilient. The agent-host environment relationship tends to be a lot more stable and stronger, and you get resistance and resilience. But when you dump everything else onto our animals and our wildlife populations, things like contaminants, invasive species, climate change, our human land uses, fragmentation, you get a loss of resilience. So we know that maintaining healthy animals and healthy populations through preventive medicine, and none of us are doctors, so we're not really applying medicine, but we're using this as a concept to say prevention is way more efficient and cost effective than reacting to the problems once they're in our face. What this means we have to do is basically pay attention. We have to really focus on the root causes of wildlife health problems instead of just running around behind the problems and sticking on Band-Aids, which is what we tend to do. We tend to wait until things become an issue before we actually step up and make the changes that are necessary. That's to some extent human nature, limited resources, and truthfully, it's quite boring to be vigilant and pay attention 24/7. And if nothing happens, that means you did a good job, but it's not that exciting, and people do tend to get a little bit more bored with that. So it's like the traditional canary in a coal mine. We think about animal health and wildlife health and environmental health really as something that is an underlying factor to human health. The concept we call One Health. Everything is connected. And the concept isn't necessarily a new concept. We've understood this, but we're trying to now apply it more in terms of our professional lives, our professional relationships. So certainly I understood that everything was connected a long time ago. Most of us have, but we have not proactively reached out to our public health departments. May not have been proactively working with water quality departments because we kind of were working in our own separate areas, and we understood that things were connected. We're in an era now that if we don't work together, we're going to not do a very good job. So this is where I'm going to take just a couple minutes to ask you guys if you have a couple of thoughts about why do we care? I took this off the Native American Fish and Wildlife Society as one of the goals, kind of one of the mission statements. I wonder if you guys, a few of you would be willing to share a couple things that might be in addition to this or an expansion of this. As to why do we care? Why do we do what we do? Why do we worry about wildlife health?
Healthy wildlife populations and natural resources in general just sort of help perpetuate traditional culture and traditions into the future for future generations.
Absolutely.
I've always gone with the concept that what affects them are going to ultimately affect us, and because they're definitely going to be affected first.
That's a great point. That's kind of that canary in the coal mine concept. Absolutely. An early detection system, perhaps. I mean, if our landscapes and our wildlife aren't healthy, perhaps we're not as well.
One of the things I was thinking is that for us here, we manage the interactions between people and wildlife and have to kind of be that intermediary on the reservation. And so we're always trying to keep an eye out for wildlife diseases and things that could pass to people. We take calls about rabies. We take CWD is a big concern because it's right at the northern part of our reservation. So just trying to keep people healthy and be on top of those types of issues so that we're managing in an effective way.
Maybe. Yeah.
Excellent. So it's pretty important for all of
Ayla's
us.
really struggling to stay awake. I was looking for a liner.
So it's important for all of us, and you guys just brought up pretty much everything that I've got in the next 3 slides, which is fantastic. We have people that are living, working, playing, hunting, fishing, certainly comments about CWD. We do have a lot of viewing, photography. Folks are making a living on lands that also support wildlife. Certainly we know that we have agriculture, we have domestic animals, we may have timber harvest, we may have poultry, we also have pets. And then a lot of us are also dealing with some feral and invasive species. I don't know if you guys heard the headlines in USA Today last January where people were being hit in the head in Florida from invasive iguanas falling out of trees. I mean, it's just, the concept is amazing. And I actually talked to a colleague in Florida. She's like, yep, that's not sensationalized news. There's really people running around that have some pretty bad lumps on their head. So while we can laugh at those, and it's great to see a headline like that every once in a while, the real world is that those things cause a lot of problems and cause a lot of headache for what we're charged with doing, which is try to protect and perpetuate wildlife populations. Our management may be really variable. In some cases, we may try to create new habitat. We may try to alter hydrology. Sometimes our hydrology altering has to do with agriculture. A lot of times we may use that to benefit and improve habitat for waterfowl. We may work to eradicate invasive species or at least contain them. We may work to reintroduce or return native species, and that's certainly an amazing and exciting thing to be part of if we get our programs to that point. There may be an imbalance in population numbers. Certainly in some areas, we know that snow geese have increased in numbers over some of the other species that were some of our wetlands traditionally. And so trying to figure out what those drivers are and should we do anything about it? Perhaps we increase our hunting seasons. Those are some things that we can do in response to some of these changes. Those are the management activities. And so some of the things that occur in our wildlife populations may be natural. Some of them may be man-made. Some of them is climate change, a natural response to man-made impacts. So it's a lot more complex and rarely is black and white. But we know that a lot of the things that we do, and sometimes with good intentions, may increase population densities, may alter the distribution of wildlife. You know, most of us are working on a computer of some sort right now, so we're using some form of energy. And again, those unintended consequences of management. Sometimes we think we're going to do something really good, like put out feed for elk, and it doesn't always work out like we think we would like it to. You know, it's part of that challenge is thinking about the individual animal versus the population. And of course, just land use changes and habitat loss. Within our field, as we try to learn more about these systems and how wildlife health and the ecological processes that are functioning to support our wildlife populations, we may do work and put on radio collars, we may put transmitters on, we may go out and do vegetation sampling. we do actually have some impact on wildlife health and on populations as well through that. There's, of course, outside influences. We're all aware of that. You know, we have mercury accumulation in some of our largest, most pristine lands in the country, and it just, there's a lot of people on the planet, and it does have an impact. So we're going to kind of drill down a little bit into wildlife disease and epidemiology, which is really the study of disease outbreaks. We really kind of look at that agent, host, and environment. If you think about that as three questions: who's involved, when does it take place, and where? Those are some three pretty important questions that we like to think about in terms of underlying epidemiology. Why? Why do we care? Because it tells us why. And is there something we can do better? Sometimes we really probably can't. But most times we can identify at least a better way to respond to something that may be out of our control. And sometimes we can identify changes in our management that can have a positive impact on wildlife health. So host, obviously, species affected. And it can be anything from herps to birds to endangered species to species that we depend on for resources and harvest. The agents of disease. There's a kind of a generalized grouping. We have viruses. We're all enjoying a temporary, hopefully very temporary for the next 6 months, run on a virus right now. Bacterial issues are huge as we start to see more about antibiotic resistance. And so those are something that we don't always hear a lot about. We do know there are some pretty significant bacterial diseases that are a problem for wildlife. Fungal diseases are kind of making a surge right now in wildlife populations as well. You look at things like chytrid, white nose syndrome, B-cell, those types of things we really do worry about as those fungal organisms are getting a good foothold in some of our wildlife populations. Parasites are something that have been studied forever and ever and ever, and we're seeing some pretty significant shifts in those across our landscapes as well. And those are certainly some things to keep in mind. There have been quite a few new diseases in humans identified that are transmitted by parasites. So when we talk about parasites moving a bacteria or a virus from one organism to another or from one host to another, those are vectors. And so maybe that virus or bacteria wouldn't show up in a new landscape unless it was for the parasites being moved around. And of course, we talk about CWD, we have to include prions. It's kind of neat. If you look at some of the early literature, there's this amazing discussion about, are viruses alive? Well, they can't reproduce outside of a host. And now, of course, you know, prions is a whole other level removed. It's an infectious protein. So it really definitely challenges our definition of life and what can technically infect and alter living organisms. And the third part of that triangle, the third part of our questions that we ask about when we do a disease investigation is the environment. Where does it take place and what are the factors involved? Are we looking at a high elevation dry desert environment, or are we looking at an environment that was recently affected by an ice storm? Are we looking at a modified landscape? Is there significant human influence or use? We looked at that picture earlier. And we identified that some of the areas in the nearer part of the photos didn't look tremendously human modified, but some of the areas in the background did. So we have to kind of think that big picture, that big scale, agricultural, riparian areas, wetlands. Are we looking at native or planted vegetation, forested grasslands? What type of waters are we looking at? A couple of folks here from the Midwest, and you guys may be dealing a lot with some of those lake or even brackish environments in some areas. So here's a question for everybody, and it should be an easy one at this point. So feel free to jump in.
My understanding is it's droplets, right, from one person to another through like sneezing or coughing.
Droplets are part of it.
Extended periods of contact.
Extended periods of contact, yes. And so that could be physical contact where somebody rubs their hand along their snotty nose and you shake their hand, or surfaces where they've rubbed their snotty hand on a door handle and you grab that doorknob right behind them. So there's extended periods of time in enclosed spaces. We're talking about that. You guys heard of aerosol? Or airborne?
Yes.
So basically, if you get down to it, droplets and aerosol are really the same thing, but in aerosol, the droplets are just small enough they float for a while, for quite a while. So that's really the only difference. And so there's been this great debate about what defines an aerosol. We know that certain diseases like measles are absolutely very highly transmittable by aerosols. They're airborne. We know that some other diseases under certain situations, certain influenzas, and they talk about aerosol generating procedures. When you're intubated and put on a respirator, that generates aerosols. And so that puts that disease organism, that agent, into teeny tiny itty bitty droplet form that floats. How do we figure this out? It's called epidemiology. This is really what it's all about. And it is a not very precise science. It is something that we do and try to look at trends. We try to do tracing. We try to rule out other possible sources of exposure. How do we know that somebody didn't rub their hand and put a bunch of snot on a doorknob versus nobody touched a doorknob, nobody touched an elevator button, but everybody who got in the elevator still got sick? So that's really what epidemiology about. It's about doing a very thorough investigation, enough that you can ask a lot of questions and hopefully rule out some things completely outright. And then you can actually look at probabilities of other things. And we care about this for wildlife health because that's how we figure out if we can do something about it, and if so, what can we do about it? If we have a disease that is transmitted fecal oral, where animals are shedding organism when they poop on the ground, and then we put a hay feeder out in the exact same spot, and the next group of animals that comes in to eat, or the same animals in the same herd, or the young animals within that herd come in to eat, then we know that we can change that. We can fix what we do with that by not packing animals tightly into an area, not treating our wildlife more like domestics. And certainly with domestic animals, it's easier for us to treat them for disease. Wildlife, not so much. So those methods of transmission, those of you that are familiar with chronic wasting disease, it's taken a really long time to figure out the methods of transmission for CWD. Early on, everybody was convinced that it was only environmental. There was not a lot of research on animal-to-animal transmission. We now have the ability to detect a very small level of prions. We can actually quantitatively detect prions, and now we're figuring out, yeah, it's in snot, it's in poop, it's in pee. And guess what? We're learning that it hangs around in the environment forever. So in the first 10 years, when a population is exposed, as it spreads in that population, it's probably majority animal-to-animal transmission. And in certain areas, we have learned about bovine tuberculosis, especially like in the upper Midwest, in deer, between cattle and deer. When you feed deer on piles of corn, they spread with a lot of nasal secretions, mucus, the snot and slobber transmission mode. But we also know that some of those diseases, including some bacterial diseases and some forms of TB, can also be spread through aerosol. It takes a long time, and that's the type of thinking we have to put on our hats, our thinking hats, when we're looking at wildlife health issues. And it's really digging into the detail, because that's the only way we're going to figure out, can we do something about it? And that's our goal, is to try to figure out, can we do something about it? Can we break that chain of transmission? Can we improve our wildlife population health? So how does the agent spread? They're often complex. Just like I mentioned, sometimes a disease will have multiple ways of spread. So then you're asking, well, what's the most important way of spread? Or what's the way of spread that I can have an impact? So in a lot of our parasite cycles, you might have a variety of species involved, including things like snails. And so figuring out these methods of transmission and how the disease makes a living in the environment will help you figure out how we help our wildlife populations have less impacts from the disease. And it requires a thorough field investigation. And like I said, for some diseases like CBD, it takes a lot of people working on it, and it takes a lot of years. Most of us that may experience a wildlife health issue on our lands that we're responsible can actually do a field investigation and work with a laboratory, and often we can figure it out. Not always, but we can often figure out what's going on and impacting that wildlife population. So one of the reasons we should investigate is that it's easy to make assumptions. It's really easy to take a look, and if you're in an area that you know chronic wasting disease is common, you think, oh yeah, that's probably COV. You might be in an area where you know bovine TB is common in a deer herd. You kind of wonder about that. So a lot of these diseases may look alike in the animal, and you really can't diagnose them very well from the outside. And in this particular case, the animal in the upper left has a brain abscess. The animal in the upper right has hemorrhagic disease. And hemorrhagic disease is a disease that's a general term for two different viral diseases that even when you open an animal up, you can't tell the difference in those two particular organisms, those two particular pathogens or agents. You have to have the lab tell you which one that you're dealing with. And the hard part is sometimes animals are infected with both. And then of course there's chronic wasting disease. So anybody heard of the term HBC, hit by car? So if you ever see HBC, that's hit by car. So a lot of us think if we find a carcass near the roadside, oh, it's just hit by a car. Turns out a lot of animals hit by a car actually have an underlying disease. Not all of them, but a lot of them do. The probability of detecting disease in roadkill, be it chronic wasting disease, be it any of these diseases, is higher in roadkill than if you're just randomly out looking at a group of animals in a field. So if you come upon this scene, you should be starting to think about what might be a problem. And we as humans tend to be like, oh yeah, I sent some samples from the lab a year ago that look kind of like this. And they told me it was botulism. It's probably botulism. Or, you know, hey, it's March and we often have a lot of birds die of cholera. Well, what about avian influenza? Right now, I can tell you at this moment, there's a pretty big diversity of avian influenza viruses circulating around the planet. So it's always a possibility. We've certainly had it here in wild birds of the United States. Certainly a possibility. We haven't seen mass mortalities with it, but I don't know that I would call this a mass mortality anyway, looking at this picture. So what do you guys think? I'm ready to chime in and throw out some crazy thoughts here.
I have a quick question.
Yes.
I tried to do a reaction to raise my hand and I think I clapped instead.
Oh,
Sorry for that.
I am so stuck at these reactions, to be honest with you. I'm just like, all I need to do is do the eye rolling one because I suck at these little reactions.
Well, I saw the
Everybody's-
hand and went for it, and then it said clapping, and I said, that's not right.
That's awesome.
But I had a question, like especially with this photo here and this scene. So is there, because I see like at least 3, potentially 4 or 5 birds on the ground, but like is there a number of birds that warrants like, okay, this like might be an issue with like or pesticide poisoning or a wildlife disease? Like what's the threshold for like, oh, I should maybe be more cautious on this, or like, this could just be, you know, like when if you're thinking about taking samples, like is there a threshold to look out for?
Absolutely. It's a great question. And there's a step down to that because some of our labs that we work with also have thresholds. So that's a fantastic question. So one of the things that we tend to tell folks on refuges is don't assume anything. Especially don't assume anything even if it's a small number of carcasses. If you haven't seen carcasses in that area before, if you haven't seen carcasses of that species before, really probably ought to ask yourself, should I go ahead and collect samples? And historically, we often made assumptions. Well, we found out last year we had botulism. It's probably botulism. It has been amazing since, was it 2006, I think, was the first year a bunch of bird flu money got sent out from USDA. And a lot of us were sticking Q-tips in bizarre places in ducks and healthy ducks, hunter, surveillance. And, we really learned a lot about diseases because we looked. So I encourage you to, the number one question of when do you collect carcasses is how fresh are they? If you have one really fresh carcass that's right next to a road, and if it's Friday at 4 o'clock, you're not gonna get that fresh carcass to the lab anyway. But you might be like, I should probably come back Monday morning because if there's another fresh carcass here, I might grab that one and send it to a lab. And I should just, maybe step up my surveillance, maybe step up my visits a little bit to this area and keep an eye on it. Now, certainly, if you have tens or hundreds of carcasses, you've got an issue that you probably want to find out about. And again, that issue, sometimes we get things that are hailstorm. We get hailstorm, just trauma. Everything was pelted by a massive ball of hail, and that actually happens pretty commonly in some of, say, our gull colonies when we have chicks. I had no idea how many chicks died from bad weather. It's part of the natural process, but then we often have other diseases that get into some of these colonies that we learn about because we go ahead and take samples. If we've got some fresh carcasses, we send them in. There's a lot of laboratories that, a lot of our state labs will provide some services. The National Wildlife Health Center will provide services. The Fish and Wildlife Service has a few labs that can provide some services as well. And especially for those of you that are law enforcement, we can help with a lot of cases. And honestly, none of us have the money or the time to collect every carcass that we ever find. But it's really about if it's super fresh and if you have a pretty good idea of a disease history of an area, or if you're seeing something that's a little bit different. The key is to know what's, quote, normal and what's not normal. And that takes time on the ground. And that takes submitting carcasses. And if you submit a carcass and it's botulism year after year after year, I still encourage you to submit the first carcass that you find each year. Yep, it's still botulism. Because it's amazing. We do find new stuff all the time. It's a wonderful question. So the second part of that is to be sure and touch base with your lab. The National Wildlife Health Center, there was some some great conversations with the whole bird flu thing, because they were saying, oh, if you only see mortality of 10 carcasses or more, mass mortality events. Well, they have limited resources as to what they can test. They're given funding to provide support. And, you know, so that was the way that they tried to give that support. We're going to help in large impact events. It turns out that bird flu in Europe almost never killed more than a handful of birds. Like almost every area that they detected bird flu in was like 5 dead birds on the shore, just like this. So it's a little bit about knowing what's normal and not normal, and then also knowing about what disease you might be concerned on. We've just kind of been told we need to be a little bit on alert for bird flu right now. So bird flu may not present with piles of dead carcasses. Cholera often does. Botulism, I mean, defined piles, hundreds, sometimes thousands, or even tens of thousands. And that's where it's really knowing what normal is. One corner of the bay on this body of water may always have a bunch of carcasses that are blown up on shore by the wind. And another corner may never have carcasses. If you find a carcass over there, man, something funky's going on. So it's a great question. And I encourage you to, if you find a fresh carcass and it's not 4 o'clock on a Friday, that it wouldn't arrive at the lab fresh, I encourage you to submit it. And if it's a situation that you don't have the resources to do it, or it's a time where the lab's not gonna be able to accept, or if the lab is not accepting any more than, or any less than say 5 or 6 carcasses, you can always bag it and freeze it. So when we freeze carcasses, we don't get that good cell, like they look at the cells under the scope, the histopathology, but they can still do an amazing amount of disease testing. They can do an amazing amount of work with culture and PCR. And so we can still learn a lot. So if I'm questionable, I'm like, oh, it's on a Friday, the lab's not going to be open. I'll just, if it's a fresh, nice, fresh carcass, I stick it in the freezer. And then that way, if you find 2 or 3 more on Monday, you've got 2 or 3 more to go, and you call the lab, hey, I got 5 dead, fresh somethings that I'm a little worried about. And so you can send some of them that are fresh, and you can send them the frozen. They may tell you to hang on to the frozen one. But that's kind of how I would go about it, and I recommend that we go about it. If we learn about those issues, botulism, cholera, some of those things, we can really actually learn a little bit about mitigating.
Thank you. That was excellent.
I was going to share, I've had a refuge that had this horrible cholera outbreak year after year after year after year. You're like, we do nothing but kill ducks. And we looked at their water management and we said, you know what? Maybe you should turn the ditch off a few weeks earlier and not have the ditch on all through December. And they did. No more cholera, seriously. No more cholera. The way they were actually flooding the landscape was attracting birds, and they were stopping short on their flyway. They weren't making their whole flyway. They were stopping short, piling up, and infecting each other. If we literally had them turn the ditch off a few weeks earlier, no cholera. So a lot of times when we do that, we can figure something out. And if we hadn't picked up the carcasses, we never started thinking about it. So it's a great question. It's a complicated question. Great question. So to give you guys a little bit of background, so I'm going to be sprinkling a little bit of information about some specific diseases. This is not in any way intended. You cannot look at this and really, you just can't make a good decision on the diagnosis. And that's the whole point. But I do want to be sure that you guys come away from this with some basic information on some of these diseases, because it helps you understand what's normal and what's not normal on your particular landscape. So botulism, it's actually caused by a neurotoxin, which is produced by a bacteria. And it's really fun if you want to get into it, the nitty gritty of it. That Clostridium bacteria is what has historically caused food poisoning in humans. There's a lot of different types of toxins. And of course, everybody now knows that Botox is some sort of a, well, and I say some sort of because I'm not that familiar with it, it's a cosmetic procedure. I'm proud of my wrinkles. And I always will be, I think. We do know that botulism has a correlation and probably is somewhat driven by environmental conditions. We usually see it associated with hot, dry, receding water lines. We rarely see botulism on really wet years that don't dry out. Now that said, In the last 2 years, I've had people contact me and said, I have pristine water conditions. We have no water decline. We don't have super hot temperatures. Sure enough, we've got dead birds, and the lab came back and said, that's botulism. And so we, in some of our areas, we're starting to see botulism outside of what we would consider that normal botulism season. So do keep that in mind. It's another good reason, if you got a carcass, go ahead and grab it. There's some research that suggests dissolved oxygen and pH may be a factor. They've been studying this disease since the late 60s, early 70s, and we still don't know how to stop it. It causes flaccid paralysis. As I mentioned, there's different types of toxins. And this is why we do this part before lunch, because it's a carcass maggot cycle. And basically, maggots are a duck's best snack. They're just the best little snacks. And so you end up with toxin-laden toxic maggots that ducks swim around like, yay, maggots, and they eat them. We don't know exactly what that concentration is, but we do know from experimental studies that some species are pretty susceptible. Some species are a little less susceptible. For those very susceptible species, just a couple of toxic maggots can be enough to put them into that paralysis situation and can actually cause death. Commonly referred to as limberneck. The two most common types associated with wildlife are type C and type E. And type E is much more common for those of you who are in the Midwest. That tends to be more common in your area than type C. Mammals are also susceptible. Like I mentioned, food poisoning in humans. So certainly, again, we talk about a difference in species susceptibility. Not all species of birds are equally susceptible, and same for mammals, but it's certainly something to keep in mind. There's been some research out there. So for decades, everybody ran around and picked up carcasses and burned them, buried them, did whatever they could with them, thinking that if we pulled all the carcasses off the landscape, there wouldn't be any more toxic maggots, which the concept makes sense. If you pull all the carcasses off the landscape, you do indeed have less maggots. What we've learned, though, is it pretty much takes just a couple of maggots. And we've actually learned, and we did a couple of studies. The Canadians first did a great study. The Canadians went out and they've actually got 2 or 3 really good studies out there. They went out and they did some work to discover how well humans find waterfowl carcasses. Turns out not so well. Then a group of folks did a fantastic study in Texas. There's a couple of photos available of people like slingshotting carcasses from a boat into different areas of wetlands, trying to simulate a bird that died from lead poisoning and a bird that died from cholera and a bird that died from botulism, knowing that, well, like lead poisoned birds might get really sluggish and slow and perhaps they'd be able to crawl into dense vegetation. Cholera birds might actually fall from the sky, so they just threw them and slingshot them up into the vegetation. And then botulism, of course, with the paralysis, some of them drown and then end up getting windblown up onto a shore, so you might have carcasses piled up on a shore. So these guys in Texas did this in a Texas marsh and sent crews of people out on foot. And I mean, it was terrible. They just found hardly any carcasses. And we knew how many carcasses were out on the ground. So I had some colleagues in the Fish and Wildlife Service in the Dakotas that were like, I've been slugging around in chest waders my entire career, dragging a canoe behind me, filling it with dead ducks my entire career. I want to know if my career was wasted. So we launched what I fondly refer to as the hide and seek study, where I had one crew of people go out and find naturally occurring carcasses during a botulism outbreak. And we put leg bands on them. We put red tags on them, special tags, not your regular bands. I ordered special tags for them. It tucked that band up under the body so it wouldn't be a real visible thing. But it was a mark recapture, so I marked carcasses that had naturally occurred on the landscape. And then we had the regular search crew come out. And I think the best they did was 19% of carcasses that we had marked that they found. And so what that means is I probably only found 19% or even less of the actual carcasses that were out there because I was a search team and then there were a search team behind me. So it turns out we really suck at picking up carcasses. So we now really recommend that, yeah, if you've got a bunch of carcasses by a boat ramp or an area where families might come or a lot of recreation, yeah, you probably want to pick them up just because they're yucky and people don't like to see them, but you're not probably going to make a difference in that carcass maggot disease cycle. People were really thinking they were going to break the botulism cycle. And the Wildlife Health Center ultimately did some research, and I think they came up with you have to reduce carcass densities down to like less than 4 per hectare. Well, with our field studies, we've learned that we're not very good at that. So I encourage you not to spend a lot of effort on that unless it's really purely for the aesthetics. Cholera is another very common disease that affects avian populations. Rapidly fatal, systemic bacterial infection. Pasteurella multocida. Pasteurella is a fascinating group of organisms. Pasteurella multocida can infect cows, but it's a different one. Turns out there's a bunch of different types of Pasteurellas, probably hundreds of them. And since we've gotten genetic techniques, we've started to figure out that some of them are a little bit more different than the others. And so we've started to rename some of these. Bighorn sheep folks will be familiar with Pasteurella. Pasteurella hemolytica and Pasteurella multocida were 20 years ago thought to be some of the primary drivers of bighorn sheep die-offs. That's actually what I did some of my graduate work on, discovering 3 things that did not kill bighorn sheep. And Pasteurella was kind of the big theory. Of course, now we, with new detection methods, believe it's probably mycoplasma. Even prior to the Pasteurella, it was called the lungworm pneumonia complex. So parasites were really identified as a problem. And we now know that all of these things can play a role. It's way more complex than we thought. But when it gets down to avian health, we know that Pasteurella can actually cause birds to fall out of the sky mid-flight. When they talk about a rapidly fatal, we're not kidding, rapidly fatal. And so certainly in other species, the Pasteurellas can cause pretty rapid mortality and some things like hemorrhagic septicemia. But birds for sure take the cake when it comes to falling out of the sky. So that's one of the things to know. You don't really ever see sick birds. You pretty much only see dead birds, or they just die while you're putting a band on them. If you were to ever see a sick bird, and I've never seen a sick bird from cholera, I don't actually know anybody who has, unless you're doing a laboratory study. You would probably see a lethargic animal that's incorporated, swimming in circles, tremors, convulses, and then dies. So cholera is pretty insane. So we know, in terms of talking about this transmission, we think about this because this really helps us figure out, is there something we can do about it? We know that there's indirect transmission by inhalation or ingestion of bacteria in food and water. There's also most certainly some direct transmission, animal to animal, from nasal discharge. And we also know that we can have some fecal transmission, and carcasses actually will seed the environment and seed the wetlands with that Pasteurella as well. So as I mentioned, the direct bird-to-bird contact is also an important part of it, but you really can't tell the ducks to socially distance. That doesn't work so well. We do know that this tends to occur late winter to early spring. It tends to occur when our avian populations are at their nutritional weakest. It's often associated with some of the beginnings of migration, and that's where we often see, as birds start to return to northern areas, we see some of these outbreaks. A lot of mortality often associated with it, sometimes tens of thousands of birds. And a variety of bird species can be affected, but waterfowl are the most common. Bird flu. It's another one that you should know about if you walk out to see some dead bird carcasses. It's a viral disease affecting a wide variety of species. This particular virus evolves with a pretty rapid genetic mutation and reassortment. So far, we know with COVID, SARS-CoV-2, that there's some mutation going on there, but it doesn't seem to be as crazy and as rapid as bird flu mutates, which is good for us. You've probably heard a lot about the names H5N1. That was the first one we were all on the lookout for, H5N1. and then H1N1, just in all of these different names. There's basically 2 cell surface proteins that those are named on. I won't go into the detailed taxonomy on that, but just know that that's why they're named. They're named on different types of the cell surface proteins. And the H5s are particularly likely to be what we call highly pathogenic avian influenza, or HPAI. We all were looking, we were doing the duck swabbing for HPAI, highly pathogenic avian influenza, H5N1, way back in the mid-2000s. LPAI, low pathogenic. So it's super important, this pathogenic terminology, it's whether or not it kills chickens, because that's what we worry about, is our food source, our commercial poultry industry. So the way this terminology, it has nothing to do with whether it infects or affects populations of wild birds or humans. So that's important. It's amazing how many people think highly pathogenic avian influenza is deadly to humans. In fact, it's usually just mostly deadly to chickens. There's not a lot of them that are that dangerous to humans. There are a few that can infect pigs, ferrets, and people. If you don't know, and you've probably read quite a bit about some of the animal research models for COVID, but we know that pigs and ferrets have very similar both cellular structure and also the actual shape and size and proportion of their lungs as people do. So that's why there's quite a few diseases, including COVID, that pigs, ferrets, and people are pretty susceptible to. And we know with COVID, we know that mink are terribly susceptible. And we also know that mink can, in fact, not only spread it amongst themselves, but they can actually spread it back to humans. And we also know that in mink in Europe, a few humans were infected. I think there were up to about 200 humans infected in late September with a novel mutated strain of this thing, of COVID. So, you know, I do kind of bring COVID in a little bit on this, because this is a great learning opportunity that we are all living through right now, because it all ties back into kind of some of our wildlife health. And why some of these things that are susceptible to humans, you need to be thinking well, you know what, pigs and some of the mustelids may be susceptible. And we also now know that cats are fairly susceptible to COVID. So each disease has its own group of hosts that it can affect. But you know, when you're thinking about some of those transmission chains and potential exposure into humans, domestic livestock, and pets, these are some things to think about. Influenza on wild bird populations, they are considered the natural reservoir. This virus probably evolved in wild birds, but most infections of wild birds don't cause clinical disease, most. This high path is actually uncommon in wild birds. It often happens that a low path form gets into poultry and then may mutate into a high path form. And then occasionally, we do know in recent years that we have found some high path avian influenza viruses, not only in wild birds, but they've actually caused disease and death in wild birds. And particularly the last round that we had here several years ago, I think it was December of 2017 that we had coming in the Pacific Northwest. We had high path avian influenza come in and ended up spreading into our poultry. We had poultry producers across the entire country affected, especially in the Midwest. But that particular strain did affect quite a few raptors, turns out. So back to our picture. And back to our 3 possibilities. You guys now know a pretty good amount about these 3 different diseases. So what are your thoughts on what you should do?
Well, it looks like you have maybe 1 fresh carcass. The rest look pretty well deteriorated. They look like different species, perhaps.
Yes, good observation. And all three of these diseases can affect multiple species. The different rate of decomposition, though, is a key. So we know these birds didn't all fall out of the sky at the same time, but we know that a cholera outbreak can go on for days and days and days. So we can say for a fact that these birds didn't all fall out of the sky out of the same flock. Different species and different rates of decomp. Still doesn't rule out cholera.
When you collect the birds, would you also want to take a water sample to send in?
I'll tell you for sure I would, and we're going to go into the detail. That's a fabulous question. Fabulous question.
It looks like the water levels might be low, so it could be a drier season.
Sure could be. That's a great environmental observation, is that we probably have some receding shoreline. Well, I'm going to give you guys major kudos, because usually I can bait people into picking a disease with this slide. You guys are awesome. You're smarter than that. So what I'm doing right now, you guys, is give you a quick break. You've just already gotten a sneak preview. So we'll take, look at my watch here. I still am an old-fashioned person with a watch and a phone. Let's take about 10 minutes. According to my watch, that puts us back here at about 10:20. I know some folks might have, actually say, my phone says 10:12. I don't know what you guys' clock says. Robert, you got a clock we can all synchronize our watches to?
Yeah, I've got 10:12 too. So let's just do 10 minutes or 8 minutes. Is that good for everybody? Just start at 10:20.
Sounds good.
Awesome. That way everybody can go refill coffee. We'll be back here at 10:20 then.
Awesome. Thank you.
Thank you.
So we're back to our picture. And I couldn't really sucker you guys into picking a bird. It's a lot of times when I do these classes, I get some great conversation about people saying, well, I think it's botulism, I think it's that. So you guys, again, get some kudos. Because we don't want to assume what it is. Like I mentioned earlier, you can't really diagnose an animal from the outside very well. We certainly can get some clues, but that's why we investigate. And the conversations that have already taken place, you guys have started a fantastic investigation of what we're looking at on this slide. So we're going to go over the techniques of a field investigation here in a little bit more detail. And the first thing we do is we document the scene. We've talked about this in detail here. That environment, where and when. Taking pictures is a great way to do it. And if you guys have ever submitted anything to the National Wildlife Health Center, they have a background and history section on their form. I do know that we send in quite a few photos to them. They're still having folks describe things and they may call and talk to you about them. It's great. I also really encourage you to take pictures. We've all got cell phones in our back pocket now. Super handy to do that. I do encourage you to think about your cell phone, and we'll talk about that in a little bit in terms of what you touch. So we might want to collect some carcasses. If there are fresh ones, certainly that are dead, that's a great option. And if there are some live ones, we may want to describe the clinical signs that we see in some of those live or sick animals. If we see lesions, we may want to describe those. And in some diseases, if there's an animal that is probably not going to make it, it's good to go ahead and get it and euthanize it to get the freshest sample possible And that gives the lab the best ability to actually figure out what's going on with the disease. And then the question about collecting a water sample, you nailed it, absolutely. If we think water quality may have something to do with it, we can always collect it. And if a lab says, yeah, no, let's just hang on to that, or you can always not submit it, but while you're out there, go ahead and grab a sample, absolutely. So, you know, in terms of a little bit of drilling down into a little bit of details, so again, we can talk about mortality events, and then we can talk about live birds. And euthanizing live birds or any other wildlife is not always easy, so we'll talk about that a little bit after our lunch break. The key in some of these carcasses, and if we collect especially fresh ones, is that the laboratory can do gross pathology, which we all joke that gross pathology is gross. It's looking at the innards of the animal. And then histology is really looking at the cells, the cellular structure, and looking at everything under the microscope. They can see fungal evidence. They can see, often they'll use different stains that can actually highlight a particular group of bacteria. And we actually have some pretty good techniques now that can even do some fluorescent labeling of some viruses. Microbiology, the lab will be doing maybe some culture isolation. They might be doing some, we call them molecular testing. That's a pretty general term for PCR. Polymerase chain reaction is actually a technique, and it essentially just amplifies or increases the concentration of something. It's usually a piece of the genetics of an organism, but someone will have worked out that, oh, it's the spike protein on SARS-CoV-2. We can detect that with confidence that we know it's this particular organism. So, and not another organism that we would cross-react with. And then we also can look at antibodies, and we don't always get blood, but if we send whole carcasses to the lab, occasionally they can actually do some antibody testing. Depends on how fresh it is. Antibodies, or it's important to note that it really just tells us what an animal was exposed to before. An animal is not going to develop antibodies to something it's exposed to immediately. But if something's been sick with something for a while, it very well may have antibodies. And again, this is all in the popular literature and the press right now with SARS-CoV. We're just now learning, does everybody produce antibodies? We don't know. It looks like most people do, but not everybody. And the amount and type of antibodies produced in response to COVID may have to do with how severe of an infection you've had. It's probably true for most diseases. Toxicology, and that's something we're collecting some of those water samples. We can really start asking some questions about the potential for toxins, be them artificial and human introduced, or there are natural toxins that can also cause problems. So the first step that you do in any field investigation is put on appropriate PPE. I have to know that everybody, PPE is personal protective equipment. So in this particular case, and we'll go on here a little bit after lunch, we'll talk a little bit more about how you make the decision of what the word appropriate means. All of us know right now, if we're going to go to a rock concert, we probably should be wearing an N95 mask. So that would be appropriate PPE. It would probably be appropriate not to go to a rock concert right now anyway. But again, that's where we ask what that term appropriate is. It's relative to the risk. We don't always know what the risk is. When we're walking out and we see carcasses on the ground, we don't really know what disease we're dealing with. We don't know what the risk is. But we can make some generally informed decisions about what is appropriate. And then beyond that general decision, we might decide to, yeah, I'm going to be a little more conservative. I'm going to be a little more conservative. And we also want to think about spreading that disease and potentially taking it other places than on this particular peninsula. So after we put on our appropriate PPE, and in this case, I would for sure be putting on at least boot covers and gloves, we might actually head out there and flip over a carcass, and it turns out it's covered in green. And in this case, we might actually, if we're walking around on that peninsula, find green-looking fish around the corner of the boat ramp, and we might find a dead bat. So again, that host is species affected. This is where recording this observation down, taking pictures, hey, it's something that's affecting fish, bats, and birds. That's not a very traditional disease like botulism or cholera. Neither of those are really known to affect fish. Don't know that there's any bird flu infections on fish, so we might not be dealing with any of those particular 3 diseases. Oh, and we go around the corner and we have blue-green paint, it looks like, in the water. So if we see something like this, we may not be dealing with an infectious disease, we may be dealing with a toxin. So again, taking pictures, documenting that scene, figuring out what those environmental conditions are, and take pictures of the different species as they're affected. And we now know that we had something that covered them in green, and we found a source of this funny looking green. In this case, you definitely want to take a water sample, absolutely. And in this case, this is one of the handouts. So these are in your email. As I mentioned earlier, they're absolutely not required. You're not supposed to use these, take these, whatever. They are free for you to use if you'd like. Obviously, it's got our fish and wildlife service contact information on the bottom. If you'd like to just keep them as a reference material for yourself, that's fine. If you'd like to modify them and use them on a regular basis within your organization, within your agency, within your department, let me know, and I can just send you the originals, and you can take my name and number off the bottom and put yours. So don't hesitate if you think that that would be helpful for you. But basically, you know, if I've got folks calling me that they've got water with blue-green paint, I ship them a kit, priority overnight, and I ship them this, how to collect these particular samples of water. So again, the lab has a certain thing that they need. Then we also want to be sure that we protect people. I've seen some amazing photos of people collecting these water samples without gloves, and they get a horrible, horrible rash. This cyanotoxin, in that there's actually some other things associated with the cyanobacteria, the blue-green algae, that aren't even technically toxic. They're just irritants. And people get really bad rashes when they stick their arms in the water on that. So I definitely recommend gloves. And in this case, for me, I'm a big fan of palpation sleeves on top of gloves. That's just me. I'm not a rash fan. So double check your PPE, talking about gloves. Get out your kit. Somebody may have shipped you one, you might have in your truck, might have in the office. Get your samples, carcasses, and in this case, you also want to collect some water. Again, this is something else that you should have in your email. It's just a fact sheet. Again, if helpful, great. In your situation, you might want to develop something like this if you have blue-green algae problems in a particular body of water. Within the refuge system, we're seeing this more and more common. We're seeing areas that have never had blue-green algae reported. We're seeing areas fairly high in watersheds as well, so that's less likely of, say, an agricultural runoff product. So we definitely encourage you to put information out there. And certainly, if you have a lot of recreation in a particular area, we do know that these blue-green algae blooms is technically a bacteria, which is tough. It's a cyanobacteria. And they were originally classified as algae, but with all our genetics now, we're reclassifying everything. It's taxonomic chaos these days. But again, whether you call it an algal bloom or blue-green algae or cyanobacteria, it's all the same thing. And they're really, really bad on dogs. Like, dogs can literally jump in the water, jump out, lick their fur, start vomiting, seizure, and die. Really bad for dogs. So if you have an area where you have recreation or waterfowl hunting and you see that, you probably want to put a sign up like this. You can work with your state, you can work with your local public health folks, whatever is best for you. Everybody has a little bit different chain that they go through. But I encourage you to at least let folks know that that water is pretty bad for dogs. I will say, I think the EPA has put together some really funny ones that have pictures of little tiny dogs that have been covered in green. And then unfortunately, they, you know, I think most of those dogs were dyed for the photo shoot, but it's not a good thing because it does kill an awful lot of dogs. So whether you are looking at dead birds, dead bunnies, dead deer, it's kind of all the same thing. You need to do the same thing. You want to document the scene? Double check your PE. Think about, what do I need? What could this be? And then I encourage you, if you're dealing with something that is transmissible to human, a bunch of dead feral pigs, mink, ferrets, mustelids, remember that their physiology is not that different from me. And so therefore that might be a little bit more of an increased risk to me. Or I have just heard that highly pathogenic avian influenza is running around in this country. Now, I have to think for sure about chickens. I don't want to take this home to chickens. I'd better at least be wearing boot covers that I can take off and bag on the scene, on site, before I take it home to my backyard chickens. Or if it's one that happens to be able to infect people, I might need to really be super careful. So again, these are all things that you want to go through in your head when you're thinking about your PPE. Get out your kits, get your samples, and certainly for carcasses, your kit's going to involve gloves and a garbage bag. We're not talking anything super sophisticated. Sampling kit, and then you're going to collect those samples.
I have a quick question, Lee.
Jump in.
In the handouts that you provided, do you have a handout that goes over proper PPE?
There should be a donning and doffing PPE one there.
Okay. And
Doffing is hilarious.
Yeah, and hopefully that's just a basic handout with a basic list of items for folks. I mean, you can always add or remove things that you feel you need to. But another question we had here real quick was algae blooms in stock tanks. Are they a concern?
They might be. Absolutely. So I don't know if any of you guys remember, and I'd have to go back to the year, but there was this theory of alien invasion in New Mexico. I am not kidding. And it seems like there was somebody on this call from Arizona, California, that perhaps you might be familiar with it. But in a nutshell, New Mexico Game and Fish got a report of some dead elk in a field. Turns out it was over 200 elk carcasses literally laying down in a field. They hadn't been shot, they hadn't been poached, they couldn't figure out what it was. And by golly, it was an algal bloom in stock water impoundments. I think there was a variety of tanks, actually. There were some tanks, there were some ponds, and they'd gotten an algal bloom in there that produced a neurotoxin. And it was severe enough, literally more than 200 elk dropped dead in a field within probably a matter of hours. So yes, maybe not. There are some red colored ones that are more common in stock tanks, but I will also let you know there's some red colored ones that are also pretty bad for fish, and some that are pretty bad for other mammals as well. So I recommend that you clean your stock tanks. I know personally at home, I get a little pink going to mine. You can actually put a little splash of bleach on a brush or on a sponge when you dump it. Really wipe it out, and then just fill it. I mean, like a drop or two of bleach in that tank, if you fill it back up, it's not going to hurt your stock, but it'll slow the growth a little bit. Some people pour vinegar in to try to change the pH of the water. I've not ever been a fan of that, but a lot of people swear by it. Of course, people swear by goldfish in their tanks, which I don't know that would work real well for me. But yes, certainly it's something to pay attention to. 99% of the time, if you're just dealing with a 100-gallon stock tank for your horses, you're probably okay. But we have seen some mortality events associated with it. And I'll also let you know that, and again, this is just us learning, within the Fish and Wildlife Service, our blue-green algae program is super new. And the way we first discovered it was we found 13 dead cows on a refuge in North Dakota. We had no idea. So sure enough, in that particular time, it was a liver toxin, not a neurotoxin. And we're like, oh, we had no idea we had blue-green algae problems. The next year, we found 3 dead cows. Guess what? That next year, it was a neurotoxin. So the same body of water, 2 years in a row. I think it was the same cooperators' cows that were affected. Felt super bad for that guy. But we actually ended up with 2 different blooms with different species of that bacteria that produce different toxins. How crazy is that? It actually turns out it's not that crazy. So definitely something to keep an eye on for your domestic animals. Just keep that water source clean. If you're dealing with tanks out on the range that you're not checking that often, keep an eye on them. And keeping some sort of a freshwater exchange is the best way to go. It's a great question.
One more thing here, Lee, if I can, just real quick. People have been asking about PPE, and I guess the main thing to take away from this is if you do not have proper PPE, document things the best you can without it, whether it's with the phone, but limit your exposure, obviously. Make contact with somebody who might have the equipment to come in and help you out, or make sure to go get it before you start processing the scene yourself.
Absolutely, and that's a great question. We're going to go into PPE requirements here in great detail. You know, that's part of that question about knowing that there's not a ton of diseases that are transmittable from birds to humans. A garbage bag flipped inside over your arm can be PPE. Seriously, that's a giant glove. That's like a palpation sleeve. If you can shove that garbage bag up your arm, reach out, grab the bird, flip the garbage bag back over around the bird, then you're golden. So the terms PPE, a ponytail holder. When I give this class in person, a ponytail holder is PPE. Seriously, you do not want your hair dragging in that maggoty carcass. I've seen so many people come out of ungulate necropsies and their hair smells like rumen content. So ponytail holder is PPE. Bug dope, we'll talk about that a little bit. So PPE does not always have to be the full moon suit, but we do have to think about how to stay safe. And if we're not confident we can do that, we absolutely want to take some pictures and then run back to the office and get what we need. Sometimes a garbage bag is good enough. Seriously. Great question. Great question. So a little bit about tools. So I will quickly just mention this particular photo was assumed to be an HBC, hit by a car. And I got this picture and they're like, yeah, we found it by the road, so it was just hit by a car. I'm like, you know, certainly possible. But we also know there's at least 2 other diseases in mustelids and raccoons and canids that can cause some really weird scratching behavior, and it often occurs on the head. Rabies, distemper. These are some things that often cause animals to literally scratch themselves, take all their hair off and scratch themselves raw. And I just wanted to mention, and we'll talk about this a little bit later, that for those of you who are doing chronic wasting disease work, if you find a deer that's dead, that has a lot of hair missing on its head, you want to ask yourself, is it possible, based on the pattern of the head and the neck, is this probably hair loss from hit by a car? Was it drugged down the road, hit by a car? Or is it possible it could be rabies? Because there's a very low risk, but it's there, of getting exposed to rabies doing CBD. Deer can get rabies, cattle can get rabies. We see a handful of cases every year. Again, extremely rare. It's something to think about. And with rabies, the secret is don't huff the carcass. Don't stick your face near the carcass. Don't open it up until you can get a mask on. And that's one of the things we'll talk about on a plague case here, a human exposure to plague, is that thinking about what that aerosol is, what that exposure is. And if you're not sure it might be rabies, leave that particular sample on the side of the road and have somebody who can come and get it that's either vaccinated or has a mask, you know, best both. So you guys should have some of these in your email. I don't actually remember if I sent you the Wildlife Health Center submission form. It is available online. And those guys have been pretty helpful in a variety of cases. They can work with your states. They can work with just about anybody that needs it. They're funded to help a lot of us. They do often have some issues with, you know, if they're being overwhelmed with bird flu, then they may not be able to accept some of those smaller cases. So just, you need to reach out to them. And they have a great website. They've actually done a really good job of reorganizing. It's amazing. They've reorganized and they are amazingly responsive now, and they're happy to answer questions as well. So this is basically, you guys have this now. And I'll just let you know, this is what some of our protocols look like. Most of them look kind of like this. We try to include some pictures, a lot of detail. They're a little formal for my taste. But it's kind of what people seem to, I've asked for feedback and people are like, yeah, it's great. So feel free to let me know if you would prefer to customize or or do something different. I would actually love to get a video protocol set together, and it would seem like this would be a good time to do that, teleworking, but I don't think I have an artistic bone in my body, so I'm probably not the person to do it, but doing little brief video protocols might be a good way to go as well. Something for you guys to think about. So we're back to our field investigation, and we're going to talk a little bit about those animals and carcasses, describing clinical signs and describing visible lesions. So what do you guys see here? Jump in.
Is that a cyst or an abscess on a deer?
That is a tentative diagnosis, which you should not be doing. Good attempt, but there's a little bit, you're thinking, so a cyst has a certain like term of an enclosed sac with fluid. An abscess is often the same thing, but it involves an infection. Do we know that much?
No, I guess not without opening it up.
Great. So thank you so much for saying that. It's awesome. That's how we all learn. As we say things, we're like, well, that's what I thought, but you know, the lab may come back with something different, but so awesome. So this is how we talk about what we see. So this is really, it looks like there's a volleyball embedded in its chest, and it pretty much looks like it's under the skin. And that seems silly because it very well could be a cyst. It very well could be an abscess, and it's probably an injury, like an artery or a hunter injury. It probably is a cyst, but we don't know that, and that's kind of where the lab will come in handy and help us figure that out. We can tell the animal's in reasonably good body condition, but we would describe this as there's a volleyball embedded in his chest, and you'll hear a bunch of snooty people say, well, don't use the term volleyball because not every person in the world knows what a volleyball is. And obviously we're all dealing mostly with people who know what volleyballs are. If I'm going to send samples to another nation, another country, another planet that might not know what a volleyball is, I will rephrase into a specific spherical lesion of whatever diameter. But certainly fine to say the lump's about the size of an orange. So I totally encourage you to do that. Somebody may come back and ask you if it's a Mandarin orange or a Valencia orange. Again, and you can say, oh, it's about 4 inches, about 5 inches. That's fine too. So we just want to know basically, is it a giant blob or is it a little blob or there are a bunch of them? So with this, we want to use our senses. We want to use our sight. We want to use sound. And yes, if we end up doing necropsies, sound and smell actually play a pretty big role. Sometimes we can actually smell an infection on an animal that's not even that close to us. Sometimes some of those anaerobic and necrotic rotting bacteria will actually, you can smell it on an animal from 20, 30 feet away. You're like, wow, I just rolled down my window and got a whiff. There's an infection going on there. So that's important to note. Next things you want to go into a little bit more detail. Does this animal appear to be mentally alert? Do I have any reason to believe that there's a neurologic impact of whatever we think's wrong with this? Does the animal appear to be alert, or does it appear to be dull? So I'll ask you guys that question. In this picture, and again, it's a limited picture, we're not seeing animal behavior in great detail here, but does the animal appear to be alert, or would you describe this animal as dull?
Alert.
Yeah, I'd say alert.
Absolutely. I'm taking a picture. It's looking at me. It's noticed me. And it's got an ear towards me. It's got another ear kind of like, yeah, making sure nothing's going to get me from my butt. But yeah, I'm paying attention to this person taking a picture of me. Absolutely. This animal is alert. Next question. This is a little bit of subtlety. Is the animal responsive? So an animal may appear to be alert where it's sitting there and it's looking around. It's aware of its surroundings, but it may be reluctant to move away from you. And you obviously can't tell in this picture as to how big of a zoom lens I use. So a responsive animal may pop up out of its bed. So it may be alert and aware of you, but it also may be responsive and respond to you. So those are two subtle differences. And they can sometimes help us figure out how bad off an animal is in terms of a disease process. Is the animal coordinated? Well, I can't really tell in this picture, but when you watch an animal walk, does it limp? And then there's always another question is, if is it limping, Is it because the leg's not working well or because the brain's not working well? And that's a tough one. If it's a leg not working well and it's a single leg limp, you can usually tell if it's a one leg limp if that head's going up and down a lot. So when you see an animal actually limp, imagine a three-legged dog. That head goes way up and down because they're compensating for the lack of the leg. An animal that is incoordinated is probably going to be uncoordinated on all fours or all of its legs, all its limbs, and maybe stagger on one leg, may stagger on another step, may just look overall wobbly or overall having difficulty maintaining its balance. So those are some of the real important subtleties to help you ask those questions about, are we looking at kind of that mental and neurologic health versus something that may be more likely to be a traumatic injury or a localized infection in one leg. That takes us kind of to the physical, back to coordination, same thing. If the animal's not coordinated, is it limping and is it one leg? We look at its gait, we look at its posture, is it kind of hunched up? If it's hunched up, maybe we're dealing with something that was gut shot. Or hunched up is often another sign that the animal's got a pretty bad systemic infection that's circulating through its bloodstream. It just feels like crap. Is it pooping? Another good one. If you see an animal poop and eat, then yeah, it's probably not quite as bad as we might think, but again, that's just a data point to collect. Body condition will tell you how long it may be suffering from a particular illness. And that helps us figure out if it's something that should be making them fall out of the sky versus something that they're probably likely to fight for weeks or maybe even months, and that the immune system is going to be very involved. So that body condition, and we talk about, there's a bazillion of body condition scores out there, lots of them developed for livestock. I tend to really try to encourage people to stick with kind of emaciated, literally bag of bones, poor, which is frighteningly skinny, fair, skinnier than we'd really want it to be, but not on death's door, not frightening. Good, yeah, medium, good's kind of middle of the road, not great, not terrible, very good. That's got a slightly roundness to it. And then an excellent, that is a plump puppy. So that's kind of, I try to encourage folks to not go into, because there's some scales out there that are like 10 scores or 12 scores, and you're like, oh, wow. There's just too many levels. A lot of people, poor, fair, good, and excellent. Just 4 scores works fine too. You're just going to describe how bony or ribby the animal is. And often that picture is really helpful. I will suggest that you take different pictures from different angles, because that really, boy, shadows and shade and angle can really tell a difference, especially on some of our cervids. Getting a shot with that hip angle can really help us figure out how skinny an animal may be. Knowing whether they've got a winter coat on can also help you understand how easy it is to tell what body condition is. Is the animal panting? Is it breathing hard? Is it wheezing? Is it breathing really fast and shallow? Or is it taking really irregular, slow, what you'd call ragged breaths? And seriously, you may find this. I mean, we've had a lot of our folks come up on deer that have been injured or elk that have been injured, bighorn sheep, you know, a lot of things that we find. And I've actually had some reports of this on some of our medium-sized mammals as well. So if you take a look at this picture, we want to talk about clinical signs. These are the signs that a live animal would display that may or may not be affected.
Lee, before we move forward, can we go back to the previous slide?
You bet. Let's see if I can make that happen. Oh, I got it done.
So
Yes.
In this situation, we had a question, would you put the deer down in this case? But outside of just that growth on its chest, in general, it looks like it's in good shape. So would you just wait for the deer and let, you know, nature take its course, or would you want to investigate further by putting the deer down and then doing a necropsy?
Now, that is a wonderful question, and that is also a value-laden question. So as a scientist, we all want to know everything about everything, and we want to know. But as wildlife conservationists, we also know that natural processes and the ability to suffer an injury or even get sick and suffer a disease and survive is super important to the long-term health of the species. So to think about it, if every kindergartner that got the sniffles, we euthanized and did a necropsies, we'd run out of kindergartners real quick. It's a wonderful question. And I have to say that in certain situations, you might be more likely to take a sample. If you are concerned about animal welfare, hunter shot placement, if you're working with some problems where you might not have, you might have a reason that you need to demonstrate that animals are suffering injury, you might choose to remove that animal, work it up, submit samples to a lab, and document its condition as that animal was affected by humans. And you might choose to do that because you need that information to support a program. Refuges, I mean, historically, we were a little more on the gather it, collect it, and learn from it side, but we've really gone a little bit more towards if it does not look likely to be infectious to the rest of the herd.
Okay.
I think it's something that's likely to affect domestic livestock or humans. And if the animal looks like it's doing okay and they survive it, we let it go.
Right.
And I tell you, so there's so much value in that. And it's been difficult. Now, I will let you know, animal welfare has to be the first call on that. So you can say, well, an animal with a broken leg may survive. I have seen three-legged bison survive. I've seen a lot of three-legged bison survive and do well. Do I, on refuges, encourage our folks to allow an animal to continue trying to keep up with a herd with a broken leg? No. I've seen it. I've seen it happen a lot, actually. Surprising. I've actually seen a three-legged bison hold a grizzly bear off for 3 days. Bear got him in the end. But so that's that value question. So ask yourself about animal welfare. Ask yourself about some needs that you may have to keep moving your program forward. If you're specifically dealing with concerns about shot placement, those types of things, then you may choose to. I would not remove this animal. This is likely, it may or may not carry that particular sac or growth or whatever it is for the rest of its life. This animal may never breed again.
Okay.
We tend to lean on the side of not being that judge. If we think it's not going to affect the rest of the herd. Now, I will also tell you that we hear from the public telling us to put that poor animal down.
Right.
Wonderful question, and thank you so much for asking it, because it's a very difficult choice, and I have to say it is case by case. Your number one question should be animal welfare, and then right with that, of course, should be risk to the herd, risk to surrounding livestock and to humans.
Okay, thank you for that. And then
wonderful question, thank you.
And we had one more question. Dewey said last year they had a hunter harvest an elk and found growths or tumors on the lungs, heart area, and the ribs, and is wondering whether or not there could be a concern as far as consuming the actual meat.
Definitely a concern. So the question is, what kind of growth? And that's where your pictures are worth 1,000 words. Now we know that things like, it's called rice breast and waterfowl, looks like little grains of rice in the breast meat of birds, is a parasite that is absolutely harmless. It doesn't affect the bird, doesn't affect people, as long as you cook it. In fact, I think you can technically eat a lot of them and it doesn't even affect you anyway. I'm not going to volunteer, but I wouldn't eat anything raw. I'm not a big sushi fan right now because we know that there's a lot of those parasites and diseases that can be transmitted. And so those are all personal decisions that you can make. But that's where when you're advising hunters, you can't look at them and say, well, that's a personal choice. You're the expert. You have to talk to them about, let's talk about those growths or those lumps. Are they abscesses? Are they cysts? How would you describe them? Are they tumors? We'll go into description here real quick. But that's where, in order to assess if it is a benign, what we would call like a benign proliferation or benign neoplasia, those are fancy medical terms that mean cells that grow out of control for an unknown reason that don't tend to cause cancerous types or other things that could spread. Yeah, if they were abscesses, little miniature abscesses scattered all throughout, I mean, you could be dealing with tuberculosis. So the whole range of that is possibility. That's where taking pictures and taking some samples, I would absolutely be concerned, depending on what you saw.
Okay, thank you.
Excellent questions, guys. So we're looking at this picture, and the first thing that we would notice is that this animal's not with the herd. So we're going to call that different behavior. You might hear the term or acronym ADR. This is a big veterinarian thing. They like acronyms. I'm not a vet, which is why I laugh at them. They like HBC, hit by a car. They like ADR. That stands for ain't doing right. It's veterinarian humor, I suppose. So what else do you notice in this picture about this animal? Jump in.
He seems alienated, left away from the rest of the herd.
Yes. So it's socially segregated. It's social distancing. Excellent observation. Excellent.
I'd say its alertness might look a little dull.
Very good. What makes you think that? It's very good observation. You're dead on correct. But what led you to that conclusion?
The ears are slightly droopy. The head isn't fully erect, and the overall, it just kind of looks a little bleh.
Excellent. Excellent.
You can also notice the squinting of the eye.
Its eyes don't look quite open. And that's hard to know for sure in a photo if it was blinking, but that's an absolutely great observation. It looks maybe a little dozy or a little dopey, perhaps. Excellent observations, guys. So guess what? You got them all. What you didn't know is it's less responsive to approach. And you couldn't know that from this picture. We do know that the elk in the back are watching me as I walk towards these group animals. And you can't know this from this photo, but I got pretty close to this animal before she actually swung her head around to try to look at me. So excellent. You guys nailed all of it. Good job. Hunched up. Hard to tell from this angle. When I got over to the side, I could tell that this animal, her hindquarters were a little tucked under, her hind legs were a little tighter under her body than perhaps normal. Almost impossible to tell from this picture, but that's another sign to look for. All right, I am seeing a yellow mark on my screen, and I'm not sure if I just did that. Do you guys see that?
Yes.
That is really weird. Oh, it went away. Cool. Maybe I didn't do it. Awesome. IT. Okay, here's another one, guys. What do you see? We're almost done for our lunch break here. You guys have been asking wonderful questions. What do you see?
Appears to be isolated. Potential not good coordination since he's leaning up against the structure. Drooped head. Body condition looks a little lean.
Excellent. Anything else? How about that hunched up? Our butt's kind of folded up under is a little funny. Excellent observations. So the photographer, this is a picture taken from a Kansas Department of Wildlife Parks and Tourism game warden. There's no zoom lens on this. This is how close he got to that animal before it lifted its head and looked at him. So that's very low responsiveness. The animal did not appear to be alert and was very unresponsive until the absolute last minute. And so there's no zoom. This guy's standing here with a cell phone, and he's actually that close to this buck before it even lifts his head. Anything else you notice in this picture?
It has some hair that seems to be falling out.
Yeah, the whole coat, the whole hair coat looks pretty bad.
Looks very malnourished.
Yes. And rough coat and malnourished, like emaciated, those two often come hand in hand. It takes nutrition to grow a healthy coat. Great observations. What else?
Drooling quite a bit.
Yes, drooling. Absolutely. Anybody have any guesses on this one? And I will encourage you to guess.
Chronic wasting disease?
Yeah. This is one of the first folks to capture a Kansas clinical, and they did euthanize this animal and submit the samples and confirm that it was CWD. But this is what a classic CWD. Now, I have seen an elk on the National Elk Refuge look not skinny like this with an okay coat, but with that much drool before because it was choking on a pellet. So you always have to keep in mind your environment. So again, I was like, I actually had called him like, guys, it's time to get the rifle out of here. We need to euthanize this animal. I've got abnormal neurologic behavior. It's twitching, almost convulsing. Its head's kind of curled around sideways and it's drooling like nobody's business. By the time our law enforcement got out with the rifle to euthanize him, he'd coughed up a pellet and was fine. But that animal was not this emaciated. So you don't always make the perfect decision, but an animal like this, you absolutely want to euthanize that animal and figure out what's going on in your herd. Absolutely. So you've heard me use the word lesion a couple of times, and lesion is just a fancy word for something that's not right. It might be damage or change in tissue. It's usually caused by disease or trauma. And in this particular picture, you can see that the quote lesion is the muzzle of this animal appears to pretty much be rotting off. And that's pretty much what's happening. So that's not something that we really like to see. So when you describe them, and this is again where pictures are great, super helpful, but it's good to know, yeah, I took a picture of this one and there's 3 or 4 more on the left hindquarter. Is there one, a few dozens? Did that deer have 3 volleyballs embedded under its skin? Size, talking about orange, a lemon, a pea versus a volleyball. What percentage of those lungs that we talked about earlier were covered or were affected by those growths? Is it 5%? Is it just 1 or 2? Or was it half of the lung was affected or 2/3 of the lung that was affected? Is it uniformly round like a volleyball? Or was it jagged edges like what we just saw on the rotten muzzle of that animal? If it's irregular shape with rounded edges, jagged edges might suggest a little bit more of a recent tear and perhaps a recent colonization of an infectious bacteria, secondary bacteria, as opposed to rounded, smooth edges might, like, you know, it's been going on a little bit longer because the body's trying to heal it. The body's trying to put down a little scar tissue and heal that edge a little bit. The edge might even be a little thickened. So the collar, how does it look compared to the surrounding tissue? Is the surrounding tissue pink and this is gray? Maybe there might be layers or rings that might be multicolored. I've seen things that are like, you know, gray to black center surrounded by yellow with pink and red. And it's good because there are some things that help us figure out what bacteria might be involved and how long the animal's been infected. A lot of times if you have gray on the center of a lesion, you might be dealing with an anaerobic bacteria. So that can help the lab figure out where to start their culture work. Distribution of those lesions. Is it on one side or is it on both? Top or bottom? Vary on the legs or does it occur all the way up? Looking at whether or not it's a smooth or rough. Is it flaky or scabby? Is it weepy and oozy? Wet, crusty, raised, or depressed? So all of these words that are great to have right before lunch, they're things that are really important for us when we're describing. And as I mentioned earlier, a photo is worth 1,000 words. I get so much crap for including a camera in this picture. We will talk a little bit more about cell phones and biosafety, biosecurity. You can take a disposable camera, and I actually have a camera that I take in the field that if it's cheap enough, I can dunk it in a bucket of disinfectant pretty much, or baby wipe it, not worry about it. I encourage you, if you do use your cell phone for cameras, it's super handy to carry along a little Ziploc. It's kind of hard to push the buttons. That's part of the problem with some of the smartphones though. So Just some things to think about as you're doing some field investigations. So we've got just a couple more slides here, and I'm going to ask if you notice anything on this animal.
Skinny and malnourished again.
Yes. And I was talking about this angle of the hips on our deer and elk. This is a perfect shot to determine that animal is emaciated.
Is it missing a tail?
It may be missing a tail.
It's discolored on the back of the head. Looks like maybe missing fur.
Absolutely. Turns out this animal was with a lightning strike. And the missing tail, so a lot of times lightning strike will actually damage the intestines and damage the nerves that serve the intestines. And we often see lightning strikes end up, if they survive the strike, starving to death because their guts don't work properly. In my career now, I think I've seen 4 lightning strike bison, one of which was killed suddenly, instantly, but 3 survived for various lengths of time. One of them was named Sparky. All right, what about this one? Yeah, they're selling t-shirts. I'm not kidding. They're selling t-shirts in Iowa for Sparky. And when Sparky finally died, 5 years later, people left wreaths in the refuge because he was popular. Sparky went viral. So what about this one? Should I be able to approach a bird this close and take a picture of it without it doing something and flying away? So this one, the bird's obvious immobile. It's not got normal mobility. The wings are spread out. Interesting though, that its head's up. So it's probably not botulism because that's usually affects the neck first. But again, we're probably dealing with some sort of a neurologic deal, whether it's an infection or a toxin. That animal's neurologically not right. And the question is whether or not it's alert and responsive. Is if I move up, you can actually kind of see its eyes are kind of half closed. It wasn't actually that fabulously alert or responsive. It didn't really try to turn its head and look at me. This one's pretty obvious. You're dealing with just an incredibly twisted and contorted body that's involving some neurologic issues, some neurologic damage that causes differential firing and contraction of certain muscle groups. And this one's pretty obvious too. That one, I mean, it's tempting to, on your clinical descriptions, just say, ew, yucky, yucky on the face. But we really want to try to describe that as a cluster of very small, round to oval shaped lesions that look like they could be growths or sacs. There appears to be some moisture associated with it. We definitely have most feather loss in that area, although you can see just a few little shafts there. And what appears to be where the eye would be appears to be kind of gray, and it progresses outward into kind of a pinkish tan with one area that's kind of dark red. Again, this is where those pictures are super helpful. And if you're not sure if this is a sparrow or an eagle, you can always stick something in the picture like a ruler or a coin to help give size. That's why you see those, because it's super helpful to figure out how big things are. And here's another one where this appears to be, we have an animal, and whether or not you're dealing with a fungal infection, which can actually cause these growths. There are actually viruses that can cause tissue change like this. The viruses are often a little bit more wart-like in appearance than this, or this could be an exposure to a toxin. This could be an exposure to a burn. It could also be the result of a freeze, although to have one ear freeze and the others is a little less likely. So again, in just describing this is a tough description. It looks like you took that little bunny's ear and crumpled it up. you know, it's crumpled, it's wrinkled. It's hard to tell what's kind of just crumpled, but it also looks like there's some crusty growths in there. We definitely have some dark red to almost black color change in some parts of it. If you would describe it as maybe parts of the area have eroded perhaps, while others of them appear to perhaps have some growth. This again, where a photo is super helpful, but do take the time, especially if you don't have an ability to take a picture, to do your best with a description. And the lab may ask you questions about it. I routinely call folks back and ask questions about what they send. So in terms of the animals and carcasses, we'll talk a little bit about the euthanasia that we would need to do. We'll talk a little bit about that in detail. On the environmental samples, I will just bring up the point that if you need to euthanize something like this, or a large angry pelican, you're going to want to know what your techniques are. And I did provide you guys with this one, that, you know, there's different ways, and we're going to go into detail on a lot of this after lunch, but I wanted to be sure and bring your attention to some of these documents and to think about that. And we'll talk a little bit more in detail about that. So we ran a little bit over. I know, Robert, were we looking at about an hour for lunch here?
Yeah. Yeah, we can do an hour for lunch. Real quick though, before we start getting into that discussion, we had one more question. And Dewey, you want to go ahead and ask that?
Awesome. I want to just say thank you guys for the questions. This is, I mean, seriously, there's nothing worse than to give a presentation and know people aren't listening. Thank you so much. You guys are one of the best groups, seriously, and I've given this to in a while.
So Dewey's question, I see that here. He put it on chat. Can those growths that he was talking about in that elk come from parasites that they get from wolves, because their population of wolves in Montana is growing? So he's just curious.
So there's an environmental data point. So that's a wonderful thing. So using the term growth, the parasites from wolves that we commonly see in elk, it's called a echinococcus. The disease is called hydatid cyst disease. And the term growth as opposed to something that would be a cyst. And that's where it's hard to describe it. So that's where we want to describe a growth looks like extra tissue that is growing outside of its normal space. So lungs normally have a clear glossy covering, almost like a super thin saran wrap, and the heart does too. There's technical names for all of that. You can just call it the glossy outer layer. If something is raised but still underneath that layer, so it's still glossy over the top of it, and if you take a knife and you poke into it, and it's got, I'm going to say it's usually almost perfectly round, and it usually has a wall that's tough and almost feels like cartilage. It's hard to get your knife into it and to pop it, and I don't encourage you to pop it without PPE on. You can see that the wall on those things, if it's one of those hydatid cysts, is usually 2 to 3 millimeters thick. It's very discreet. And the lung tissue right next to that wall will look normal. So you have this very discreet, teeny tiny thing it may be the size of a grape. It's got a very distinct, thick, tough, almost cartilage-like wall. And if you stick your knife tip in it, usually clear to slightly milky fluid will come out. And if you look close, you can usually see teeny tiny white flecks floating in it. If that sounds like what you saw, then there's a good chance that those are the echinococcus parasite. If what you saw is more like a growth, which may have broken through that glossy, or is not a perfect round grape shape and not a discrete capsule filled with fluid, then you could be talking about abscesses. You could be talking about a cancerous or a neoplasia growth. And I have seen cancers in wildlife before as well. The hydatid cysts, we most commonly find them in the lungs. I would say second most commonly find them in liver. I don't know if I've ever seen them in the heart. I'm not saying it's not possible with a severe infection, but I don't know that I've seen it in the heart. So does that help at all?
Yes, he said that what you're describing is similar to what he saw in his photographs, or has in his photographs.
Got it. Excellent. So that's good news then. So we know that those are, well, there's some evidence that certain stages of those, like if you were to go and eat wolf poop, you could probably be infected. Yeah. I don't recommend it. I mean, you guys have already know me enough now to know that things like, you know, don't lick your fingers, don't huff the carcass, and don't eat wolf poop are some of the take-home messages of this course. That's all part of the PPE. Don't drag your hair in the rumen. So basically, you know, there are, if you were to probably drink a gallon of that fluid, there is some evidence that very large exposures to the fluid in those cysts, I think a very tiny proportion of those could be infective. I was actually told, and when I was trained on this, that they're not infective, that you could drink the fluid out of those cysts all day long. Told by a few people like, yeah, we're probably not going to say you could do that anymore.
Right before we eat lunch.
Yeah, exactly. Yeah, I'm a lot of fun. So it's always fun. But, you know, I personally, when I'm working in an area where I know those, I usually put on safety glasses. And I just keep my mouth away from them. And I'll usually pop one to be confident that's what I'm looking at, because you can get other abscesses that are bacterial infections that look similar. And actually for fun, for those of you who are in Montana, brucellosis in a joint can actually look almost exactly like that, but usually the whole joint is affected and has a bunch of fluid and white chunks in it. And of course with brucellosis, you don't want to get your face near that either. And with brucellosis, that's another one you don't want to rub your face on it. So but yeah, if that sounds like that's a good chance that's what it is. Low risk for humans, but I still don't encourage you to roll on the carcass or eat wolf poop. Fabulous questions, guys. Anything else? Well, let's maybe jump back online here then in about an hour. Does that work for everybody?
Works for me. We're on your schedule, so you want to just call 12:20?
12:20 sounds perfect. Thank you.
All right, guys. We'll all jump back on in an hour. Enjoy your lunch.
Okay.
So everybody enjoy their lunch and think about describing your lunch really thoroughly. It's something that won't leave you as you go. So I want to talk a little bit more about drilling down into some of these topics. This morning we did kind of a big general overview. We'll drill down a little bit into some of the issues about euthanasia, PPE, some of those things, a little bit more of the detail, and several things that we've already talked about. Let's see if I can get my slides to advance. There we go. So, you know, we talked a little bit about animal welfare in terms of making decisions on whether or not you want to collect an animal that may be unwell, be it perhaps from a traumatic injury or possibly a concern for disease risk for the rest of the population. So what is animal welfare? And certainly, I don't know if, can you guys see the Zoom menu on your side, or is it just showing up on mine on top of the slide?
I don't see it.
I don't see it either. Which menu are you talking about?
So my little Zoom pop out down menu is on top of my slide. As long as it's not on top of yours, that's fine. I didn't know if it would show through sharing the whole screen or not. Excellent, it's not. So everybody talks about animal welfare. We all kind of have this basic understanding of what it is, doing the right thing for the animal. You know, and certainly there have been a lot of conversations over the years, and animal welfare conversations can be taken very far and be used politically motivated. They can be used in ways that are maybe not necessarily focused on the animal. One of the definitions that has been commonly used has also been termed or phrased as the 5 freedoms, and it really focuses on the animal itself. And the first of which would be freedom from hunger and thirst. And the second is freedom from discomfort, freedom from pain, injury, or disease. So notice that, you know, discomfort is identified as separate from pain, injury, or disease, and there's a little bit of different nuances there. Freedom to express normal behavior, and that's a tough one. In our human modified world, sometimes it's hard to know what normal behavior would be. We don't have as large landscapes without fences as we used to, so we don't necessarily know what some of that normal behavior would be for wildlife, but we can do our best estimate of what that might be in terms of how we're limiting its potential behavior to the extent that we know. Freedom from fear and distress. This one's also pretty important, addressing the psychological condition of the animal. So, I'm gonna jump out right now. What is wrong with this picture? Make sure everybody is not taking a post-lunch nap. What's wrong with it?
The dog shouldn't be right there.
Absolutely. Absolutely. Should we really kind of be posing and holding wildlife for the cameras? Probably not to begin with. There's certainly a balance of needing to communicate and share what we do and try to communicate the value to the public. But certainly in this situation, probably not a good option there. From my perspective, I have been very, disappointed with the amount of media from all organizations and agencies that shows a picture of a person holding an animal or next to an animal. I think that's part of the reason that we have people in Yellowstone putting baby bison in the back of their Subarus or in the back of their Toyota Highlanders, because people don't really understand that wildlife should be wild and that they're not to be picked up and handled. And there's certainly a lot of that out there on a variety of media platforms. But this is an especially difficult one. So we'll go on to what does euthanasia mean? Officially and technically, if you break it down into its Latin parts, it means good death. And how that is defined and described as appropriate to different species in terms of the Animal Welfare Act is something that we'll talk just a little bit about. Keep in mind, though, you know, the animal welfare and how those affect our euthanasia protocols. And it kills me that we have to have a list of unacceptable methods because, you know, when you read something that says, do not place this bag over your child's head, it's because somebody did it. The same goes here. These methods have been used. And it's a sad thing, but they have been used and not acceptable. Certainly, at least not acceptable under our Animal Welfare Act and not acceptable under what's considered general guidance for promoting the 5 freedoms. So then we get into the acceptable methods, and that's really tough because we don't all have access to the chemical methods of euthanasia. And certainly we're not going to be able to use injectable agents on most free roaming wildlife. So mechanical gunshot, that's something that we certainly use commonly. Cervical dislocation, you know, wringing the necks of ducks, decapitation. Those are some methods for different species that we are able to use. Ideally, if we had the ability to, we would do a one chemical to reduce awareness followed by mechanical, but we often do not have that opportunity in free roaming wildlife. And you know, the Animal Welfare Act talks, it was specifically set up for laboratory species, and it's been expanded basically to all mammals. Different organizations can expand and have expanded it further to really address some of those issues that you're dealing with in feral species and invasive species. You know, pythons, how do you deal with that? The which is best question boils down to the species, down to the environment, the materials that you have on hand, safety for people, certainly. We're not going to walk up to a bison and try to give it an injection. And your team experience plays a role. If you've got folks on your team that are not comfortable with using certain methods, you should not be using those methods. Or you need to find different folks on your team. And that experience, comfort level, and training is super important. And we need to maintain the professional standards for our profession, for our department or our organization. And the truth is that everybody has a camera in their pocket now, video. I saw something the other day about some person in Texas, sores on her feet are now being used by anti-vaxxers in Europe. Anything you put on social media for sure is going to be used. But even when we're out in the field and when we're out, if there's other folks around, even just driving down the highway, you may very well be filmed. And just to keep that in mind as we make our choices and make our decisions. This is one of the questions that I get very commonly. How do I know when it's dead? Lack of a heartbeat is certainly a good sign. Can you always hear the heartbeat on some of our larger ungulates? Not always. Lack of breathing, that's another good one. And that is tough because sometimes you will get sporadic breaths, we call them agonal breaths, that may be the result of neurological firing from the brainstem. There's no awareness or no consciousness remaining. But if the brainstem is still firing signals into the lungs, then the heartbeat might actually persist quite a while after breathing even stops. The heart kind of has its own electrical system as well. So those are good ones, but they're not always the easiest to tell. My favorite is you don't get a blink reflex on the eyeball. That's something, if you've euthanized an animal, you can usually do. And if you do get a blink reflex, you need to repeat the euthanasia procedure. And so that really is testing that neurologic connection from the body to the brain. This one's kind of obvious. Rigor mortis is setting in. There are a few diseases such as anthrax where rigor mortis does not actually really happen. So that's a little tough one to say is a universal, but it is also obvious, you know, in many cases. So we're kind of back to our same picture here. We've already talked about this, and we want to talk a little bit, I want you to think about this image and this situation, if you were to find something like this, in terms of safety, because certainly we can have our investigation and figure out what's affecting a wildlife population and down to the individual animal level, but we also have to really be thinking about our safety. So when we're collecting animals and carcasses or environmental samples, we have to remember that there are diseases out there that are really dangerous for people. I don't know if you guys saw this, we're more than a decade ago now, but many of us remember hearing about it. And it's interesting because in this particular article, it talks about this biologist who brought the carcass of a mountain lion to his garage and did a necropsy. Many of us question whether or not having that cat draped over his shoulders, basically. He had the animal wrapped around kind of the back of his neck, draped over his shoulders, and hiking out with it. Because as you're walking with a cat draped across your shoulders, you're doing some compressions on the carcass. The rib cage is being compressed. So that lion dead lion, that carcass, may have been producing plague aerosols while the biologist hiked the carcass out. He may have been infected before he ever opened the carcass. We talked a little bit about rabies, again, a pretty low risk of exposure, but there is a slight aerosol risk. So these are just things to keep in mind. Think about your safety, the safety of your family, safety of pets and other animals that you may have at home. It's important to protect the community, protect local agriculture. We all like to eat. Provide the best possible materials for the diagnostic lab. So that's something that we think about. If the carcass is like, not sure that's good enough for the lab, don't grab it. Don't expose yourself. So be confident when you do decide to make that collection that it's going to be a good sample. And if you're not sure, contact somebody who has done this. Contact some folks with some experience. When you're talking about these, we've got these funny looking things on the lungs. Take some pictures, send them to somebody, get some information, and bringing that topic up on this call is awesome. I certainly can't do a remote or virtual diagnosis, but it sounds like that you have a pretty good idea of what you were looking at, and now have some good descriptions to help ferret that out, and perhaps want to take some samples on the next time that you encounter that. So, we hear about some diseases. We might see an animal, and that's an emaciated animal. We talked about lightning strike can actually damage the nerves that serve the intestine. There can be genetic defects there. But there are some diseases like Johne's disease. There is now a pretty conclusive link between the bacterium that causes Johne's disease in cattle and bison, and occasionally in other species like elk. Johne's disease can be found in goats, with inflammatory bowel disease in humans. This is part of that epidemiology. So they noticed that the group of humans that were most likely to have Crohn's or other similar inflammatory bowel disease syndromes were dairy workers. And we know that Johne's disease is basically a fecal oral, and a lot of the dairy barns are pretty highly contaminated with feces. Milk collection, those types of things can end up with that transmission of disease within that dairy herd pretty easily. And it turns out dairy workers were some of the most commonly infected. We also know that this particular organism a very few of the bacterium can actually survive pasteurization. This is a really good reason that unpasteurized milk can be a risk, especially for folks who are immune compromised. This one's really recent. This was right about the start of the pandemic. I work a little bit with the lab director at this lab, and they do chronic wasting disease is one of the huge things they do. That's what their staff are all pretty much all gathered in a lab for. And they had 5 of their biotechs test positive. And they are all wearing appropriate forms of PPE or respiratory protection. They are meeting their lab safety protocols. So for the guys on the left that you don't see a mask, those are called PAPRs. It's a powered air purifying respirator. It's kind of like scuba gear on land. And it basically puts air across your face and pushes it away so that air isn't coming in through the room. And it's got a HEPA filter on it. It's got a filtration system. So it makes sure that the air on your face has been filtered. There's a protocol for changing the filter. So they had this huge outside safety room. Nobody can figure out how these guys got it. They can't even know for sure they got it from the lab. It's impossible. In Michigan, we know we have, bovine TB in cattle and deer, but this is not that either. So it's kind of like, how did this happen? We don't know for sure. Epidemiology has not actually figured that particular case out, but it's important to keep in mind. So it boils down to everything's connected. Everything is possibly a risk, and we have to kind of think about that. 75% of emerging infectious diseases in humans have an animal origin. And we talk about diseases that can affect the health of both animals and humans and can be transmitted between the two as zoonotic diseases or zoonoses. You'll see that term zoonotic pretty commonly, and of course, that's something that we're talking about right now. Here's a kind of just a partial list of some of the things that we might encounter with wildlife. And of course, with COVID-19, that one for sure came from animals. And we now know that it can for sure be transmitted from animals to humans, humans to animals, it can actually mutate back in those animals and then go back to humans. So it's pretty free flowing between humans and some species of animals, specifically mink, probably ferrets, a small number of cats. And there's a lot of ongoing studies on whether cattle can be infected with. Same for whitetail deer. There's a lot of research going on, and I suspect that we will see a ton of publications hit our journals here in the next 2-3 months, because a lot of folks have been working on this to try to identify what species are not only susceptible to COVID, but might be able to shed virus and not only infect their herd mates, which could have a huge impact on our agriculture, but also infect people. So biosafety is the concept of protecting yourself and other folks around you from being infected with disease agents. You need to assess the risks. Within the Fish and Wildlife Service, we do something called a job hazard assessment or job hazard analysis. It often ends up being something they refer to as a tailgate safety briefing. Those are some of our kind of little terms for it. I don't know what diversity of other terms that there are out there, but you kind of get the concept. It's assessing the risk. It's asking the question, how much of a risk is this to me? And can I do something about it? Using the right people for the job. If you have folks on your team that are not well trained, get them trained. If you have folks on your team that are incredible germaphobes, I'm kind of a germaphobe, but I'm kind of comfortable in a maggoty carcass, not so comfortable around people. I could so not be a dentist. To me, the inside of another person's mouth is terrifying, but I'm okay with maggots. We all have our own comfort zones. We all have our own experiences that will drive that for us. So assuming that most of you on this call are probably similar to me in some of those respects, we're all wildlife folks, we probably come close to at least passing the maggot test so far in our careers. If not, have fun with that. Wearing appropriate PPE. We'll get into what that word appropriate means. Now this one's fun. Wear PPE appropriately. Then I'll get into more detail on that here in a minute. Create a safe work area. This sounds so cliche. It really does. But I can tell you some really fabulous stories about people who've been hurt because they left their backpack in the wrong spot. When we were in the field, they left their backpack on a spot, and you know, a bison got hung up in it, and then somebody got hurt trying to get the backpack off the leg of a half doped up bison. Certainly a safe work area involves making sure that everybody's informed what you do and don't do. Don't be injured if an animal has been chemically immobilized or darted. And if the animal pops up quicker than you expect, do not jump on it, because you're going to hurt yourself and possibly the animal. And I've seen that happen quite a few times. So a safe work area includes more than the obvious slip, trips, and falls. Don't lean a ladder over your workspace. I've actually, I have some jokes about a friend who I was actually on the phone with who did the proverbial old-fashioned file cabinet drawers. And if you open 2 of the top drawers at once, the file cabinet will tip over. I was actually on the phone with a person when that happened. And I mean, it wasn't funny, but it was funny because none of us, we've all read about that. File cabinet safety. I was on the phone with a person when they had a really heavy file cabinet hit them hard in the chest and they were bruised for a long time. So it covers the gamut, and you want to think about it from head to toe. Using the right tools for the job. I do not recommend using a butter knife to do your necropsies. Many of us that started early with chronic wasting disease sampling actually did use butter knives to take out obex. It was before we knew that retros were a good sampling tissue, but certainly we customized our butter knives and made sampling tools out of them. So using the right tools for the job will make it not only easier, but safer for you. And take the time to do it safely. That's one of the things I was talking about. If it's late on a Friday, you're not sure about the quality of the carcass. If you're exhausted, if you don't feel well, if you're sick, you're tired, you're hungry, you're late, think about your safety. It's super important to get that data and get those samples, but if you're not confident you can do a good job and a safe job, don't do it. All right, does anybody know what this is?
Isn't
A
that
cootie.
ant in my pants? Oh no, that's
Isn't it a cootie?
yeah.
Yes.
Yeah, it's a
Who
cootie.
said that? Who said cootie?
Holly.
Hi, Holly. You're awesome. This is the Hasbro Brothers, I think, branded cootie toy. Does anybody know where that comes from? Where does the cootie? Does anybody remember? And I'm dating myself here. The old childhood taunt. You've got cooties. Has anybody ever heard that? And do you know what it comes from?
Isn't it lice?
It's probably been used for lice. It was originally polio.
Ah.
Yeah. This cootie's got a long history. I wonder, I'm kind of hopeful we don't, get a newly branded COVID cootie toy. But, the whole goal of this is to think about not giving yourself exposed to cooties and not taking cooties home, because really and truly, the germs are going to get most of us in the end. There's this really amazing study where a bunch of Park Service employees in 2 different parks volunteered to get their blood drawn. And at the end of that particular phase of the study, 3/4 of those employees, turns out, had positive antibodies indicating past exposure to a zoonotic disease, a disease that had come from animals. A year later, they got blood again, and 16% of those same employees had new infections that had not been identified in the first blood sample. So that proportion of people, that gives you a little bit of an idea of an exposure rate in that particular group of people. The worst thing is that most of them missed work because they felt like crap. They were sick. So it's not just like random, oh, look, I have a unique antibody. Gee, didn't know that. And certainly for those people who experience asymptomatic COVID infection, yay for them. There's enough of us that are not likely to or will not. That's certainly a concern. But when we look at our wildlife folks, when we look at all of our professional colleagues and all the things that we do, we have to really be aware that we do have some exposure risk. And so and this is just a partial list. Some of these you're going to be like, oh yeah, heard of, maybe not. But keep in mind that some of these are more common in other species. So birds, of course, we know that several strains of bird flu can be infectious and cause problems for humans. They are rare, and you should be notified by someone official if they've detected bird flu in the United States or in your region. When we get bird flu that affects our poultry, USDA is all over it and it hits the press. A lot of our public health folks are notified. But if we get avian influenza that affects humans, just like our swine flu several years ago, we find out about it pretty quickly through both official and unofficial chains. Salmonellosis, everybody's familiar with that. Salmonella is a normal part of a bird gut and is a normal part of bird poop. So it's not unusual. It's quite common, in fact. And many species and mammals are actually susceptible to salmonella as well. Rodents, a lot of you guys are familiar with hantavirus, leptospirosis, plague, and tule or tularemia. Carnivores and bats are certainly a concern for rabies. Brucellosis, since about half of the folks on this call sound like they're from Montana, I'm sure you guys are pretty familiar with brucellosis. It is spreading in elk, and it is a great concern. I've often wondered if brucellosis is not a good risk predictor for spread of chronic wasting disease. It looks like in Montana anyway, CWD may have moved faster through our elk and deer herds than brucellosis has. But certainly, we know that we had brucella coming into some areas in south central Montana across the Wyoming border before CWD was detected there as well. So we can kind of pay attention to some of the movements of some diseases in some of these groups and start to think about how we might anticipate other disease movements. We talked a little bit earlier about insect vectors and parasites. And certainly we know that quite a few of these diseases, triple E, Eastern equine encephalitis virus has been in the news quite a bit lately, and it can affect people. It's been pretty common, more common on the East Coast. Tick-borne diseases, Lyme disease, you know, that's kind of the stereotypical red target. Interestingly, different ticks on different continents have a different probability of producing that red target. So it's not a guaranteed sign for that. And there's other signs, and encourage you to get tested if you are concerned that you've had an attached tick for a while. There's not been a lot of endemic Lyme in Montana. We do have some of the other tick-borne illnesses that affect humans, but I tell you, with my job, I've traveled some, and boy, I have done my best to try to keep ticks off. And we'll talk about bug dope being a bit of PPE here in a second as well. Tularemia can spread by respiratory route as well. That's something to keep in mind when we've talked about not hovering over our carcasses and breathing deeply. Plague is a very stereotypical classic flea-borne illness. It can also be spread by the respiratory route, as we just talked about with the biologist in the Park Service who carried out that mountain lion on his back and across his shoulders. So I'm going to give you just a little bit about each of these diseases. Plague, most mammals are susceptible. That said, fleas actually turned out to be pretty picky. So the flea species that are most commonly involved in the plague cycles are not the most likely ones necessarily to jump on your horse or your dog. Although your dog, when it runs out and sticks its head in a prairie dog burrow, is going to come up with some of those fleas. And then when your dog crawls in bed with you, guess what? That's the most common way that humans are exposed to plague. I suspect that running around in sandals in prairie dog towns is also probably not the smartest move. And trust me, I've seen that a lot as well. Primarily Western distribution, as you can see with the map. We talked a little bit about pets. And I encourage you, if you find dead prairie dogs, to close the area to people. And there's all kind of signs. CDC has signs. This particular one I worked with the Colorado Department of Public Health on. They're the one that provided the giant flea clip art. I don't know what you refer to that. Is that a flea logo? At any rate, work with whoever you need to get the message across. Certainly on refuges, we have discovered that we put a volunteer housing area smack in the middle of a prairie dog town. Nobody was hurt. We found a couple dead dogs and we're like, hey, that's probably not the smartest thing. So we moved that housing location. In the meantime, encourage people, wear bug dope, wear pants, and don't wear sandals. Tularemia, again, another one many mammals are susceptible to. We're talking about this because these are the most common diseases that we might find that present a risk to us, to people. Tule is more commonly found in rabbits. There's also a wetland cycle in moose and beaver. And, you know, I've actually gotten calls from fisheries crews that are working, doing electroshocking in streams. They're like, man, we keep coming on these beaver dams with dead beaver. I'm like, oh, that's not good, because tularemia can actually be found in the water. And so you probably don't want to necessarily be working in that particular reach's stream. And another, ticks and biting flies are pretty common vectors in transmitting Tule. One other common exposure route is do not run your lawnmower over dead bunnies. It really can be a significant source of an inhalation dose and exposure to tularemia. And tularemia that's goes down into your lungs is a bit harder to treat. Rabies, you guys are probably pretty familiar with this. Most people are, most wildlife professionals are. Neurologic disease, it is almost always fatal in mammals, and I believe the 1 or 2 humans that have survived wish they hadn't. There are geographically distinct reservoir-specific variants, and so you can kind of look at this map, and this is a little bit older data. The variant doesn't shift much at all. And I actually did a lot of digging, and in the last several years haven't seen these maps produced as consistently. This is out of a CDC publication, actually. And depending on where you are geographically is what the most common variant is. Now, what that doesn't mean that the other species may not carry it. There's different variants that are more common in that particular species, but absolutely, again, these other species, you may be in an area where skunk variant is common, but you're absolutely have a good probability of finding rabid bats. Again, you know, the biting from pets, contact with tissues. This is where if you happen to run across the one deer in a million that has been infected with rabies that year and you're doing CBD and you're not wearing gloves, that's certainly a possibility. And then inhalation is another concern as well. And we talked a little bit about some of that hair loss as one of the signs of rabies, potential signs of rabies in cervids. Generally, there's dumb and furious. You guys, I'm not going to go into great details on that. And this is where really, you know, you want to think about this when you're asking, oh, it's probably just hit by a car. Yeah, just be careful with it and don't make the assumptions. A lot of these species, when we're talking about things like rabies or distemper, many of our public health organizations want these samples. So there may be a state Department of Health or county Department of Health that may be interested in, and maybe some of these departments actually get some funding to track what rabies is doing in their area. Most of our public human health folks do not like to see rabies get to the point that people are getting exposed. They want to know that they've got rabies active in their geographic area that year, and to be able to get the appropriate messaging and warnings out to the public. So I do recommend for the species that have potential for plague, rabies, and tularemia, those three especially, and partly because, and we can ship them with FedEx, and if you don't find anybody interested in it and you really want to find out, that's where you start reaching out to other labs like the Wildlife Health Center. There's quite a number of labs, some of the state university labs. So like here in Montana, the Montana Veterinary Diagnostic Lab. The Montana Vet Diagnostic Lab has been fantastic about accepting samples that could be tularemia or rabies. You got to get them heads up. You got to be sure that that package is packed correctly. We'll go over that briefly. But it is certainly something, if you want to know, there's ways to get it done. Not something I recommend that you if it's small enough to fit in a box, don't cut it open, and that's why. We won't talk about feral swine too much. There's a whole lot of information online, but let's just say that they certainly, not only in some areas, have the potential to play a role in disease transmission, but we know that in some areas with intense agriculture, they are playing a role in disease transmission to humans. Feral swine can certainly contaminate fields with salmonella. That's been pretty clearly documented in a few cases as well. Interesting, a lot of folks really thought that there might be pretty high concerns with trichinella, and not so much. It's kind of interesting, and whether or not that's because it's been nearly eradicated from our domestic production or our domestic pork production, or if that's just not something that this particular animal tends to host. Not sure about that. State and USDA management, certainly a variety of jurisdictions involved here. But yeah, definitely something to keep in mind if you're handling feral swine. African swine fever is not here yet. Most wildlife health professionals that are in the foreign animal disease world believe it will be. At the rate that it's spreading on the other side of the planet, it's probably a matter of when, not if, it will get here. So while people don't appear to be susceptible to African swine fever, we may very well be a method of transmission. And that's one of the really important things. This particular virus is pretty darn hardy. And that's where wearing those boot covers and taking them off and bagging them before you go hike somewhere else is pretty important to not spread it. So biosecurity is an approach to prevent the introduction of disease to new locations. So we've got biosafety to protect us, biosecurity to protect everything else, basically, is what that means. Again, we're protecting our family, friends, neighbors, pets, livestock, food supply. Just the goal is to keep it from spreading. We saw this picture before. Like you to comment on this picture. You might see it again. What are your thoughts? Other than yuck.
He needs a toothbrush.
The unfortunate thing that you're seeing between those teeth is that's not just stuff. That's rotting tissue. His gums are rotting off. Toothbrush won't fix it. That's actually a disease. This is, just so you guys know, is what hemorrhagic disease looks like in some of our bovids. Hemorrhagic disease is common in deer. It's so common in the Southeast United States that animals in the Southeast don't tend to have much symptoms. As we move further north and further west, animals don't get exposed to it as many years. And so their antibodies and their resistance kind of will peak and then kind of wane. And when that resistance in the herd, we've all been hearing about herd immunity with COVID. It's certainly a real thing with wildlife and exposure, that we see more severe disease and more significant lesions and mortality. And this is what, if you were to describe it, this is where the tissue's been eroded or ulcered. I mean, you could almost reproduce this by pouring acid on those tissues. So that's kind of looking at those clinical lesions there. So if you get something like that in our domestic food supply, that's really bad. And it does happen. You know, hemorrhagic disease does affect our domestic cattle. Some years not so bad. Year 2012, pretty bad. We had a lot of cattle mortality, especially in the Midwest. And you get something like that in a feedlot, not that great. So something to think about. Same thing, you thinking about one backyard chicken, a small backyard flock, and you get that same disease, that avian influenza or another disease in a commercial production facility, you're talking millions of dollars a loss, millions of dollars a loss. It's unfortunate that, you know, regardless of how many of us are hunters or fishermen, that many of us are still relying on some commercial feed production, and we do certainly need to keep that in mind with our biosafety protocols, to not be that person that spreads it and introduces something into agriculture. And so if you think about biosafety and biosecurity, how to infect yourself with a terrible disease and how to affect everything else and everywhere else with it, the good thing is that it's a huge overlap in the preventive techniques. So they're really the same thing. The goal is to contain it, is basically contain it to the site you're on, and that's it. We've already talked about all of these different disease exposure routes. This is just a little table. And you don't need to worry about exact diseases. These are just examples, things to think about. And we talked earlier about how at this point, COVID, you know, SARS-CoV-2 looks like it can be at least 3 of these. There has been some question, and I think there have been some folks looking at research. Can it be mosquito-borne? And so far, my understanding is that preliminary research suggests that COVID cannot be transmitted by biting insects. So, you know, that's a good piece of news. But many diseases are transmitted by more than one transmission route, which makes our field investigation important to figure out what's going on and to help figure out what disease pathogen is it, what's the agent, and knowing the transmission route, what can we do to slow it down or break that cycle? So with biosafety and biosecurity, we talked about a couple of things that is assessing the risk. What species is it? We certainly know that pigs, the mustelids, are more highly related to people. We know that if bird flu is known to be circulating in the United States, we need to be extra careful because we don't want to introduce it into poultry. And if it's a zoonotic avian influenza, we might not be going out there to collect those samples at all. We may be saying 1-800-USDA, I don't want to pick it up. You come do it. I mean, we all have to get down that road when it happens. A few places have experienced that already. But certainly, you know, the United States has not yet experienced an avian influenza that can infect people. And those are where you really got to get your respirator certifications down. And that's something we'll talk about here in a minute. The season, you know, seriously, there's a lot of other risks associated with season. If your hands are too cold to handle a necropsy knife safely, then it's probably not a good choice. Been there, done that. I'm like, nope, not going to do this anymore unless we can get in somewhere out of the wind. Because doing this at 26 below is not smart. Not only am I going to cut myself, but this is a brucellosis infected animal, I'm going to infect myself with brucella. Nope, we need to find a garage. We need to find something to help with this. And take a look at the clinical signs and to try to get a ballpark idea. Be like, yeah, I'm not going to diagnose this ahead of time, but you know, I'm really thinking this looks like a gunshot wound. This looks like an animal that survived with a traumatic gunshot injury, and I'm probably not an infectious disease risk from that. Environmental conditions, we talked about temperature, wind, the slips, trips, and falls. I have seriously seen people almost fall face first into a carcass. Not good. So keep that in mind. Sometimes people will want to protect the floor. Oh, we're going to bring our carcass into a garage. We're going to do the elk necropsy or the CWD sampling in a shop. And we don't want to clean up that cement floor. So we're going to cover the floor in plastic. Guess what? Plastic is slippery. If you get a little blood on your shoes or on your boots, it's very slippery. So I caution you to think about these things in terms, not only in terms of ease of cleanup, but also the safety to the people. If you don't feel well, you're not on your game. And if you don't feel well, and if you have a human influenza and you handle a dead bird that has an avian influenza, you could absolutely be the mixing vessel for the next pandemic. If you don't feel well, don't go picking up dead carcasses. Don't go out there. Don't be doing the necropsies. Stay home. Some of these pathogens do have a pretty high rate of genetic reassortment, and don't be the mixing vessel for that. If you're tired, and if you're like me, like food and water is huge. I have to eat pretty frequently, and I have to have a lot of high protein. I'm like constantly eating nuts and jerky. And if I don't get that, then I am not going to be safe doing work in field work or necropsies. I'm going to stab myself. I'm going to stab somebody else. If I have to draw blood from live animals for anything, I do not drink coffee. I don't drink caffeine because I'm going to probably draw blood from my coworker. Not a good choice. So be aware of your personal situation, that moment, that day, and make good choices. We talked about the right people for the job, comfortable and trained. If this photo freaks you out, you might not want to be the person doing it. I have seen in my career some vegetarian biologists being taught to do necropsies. And, you know, that's a personal situation, and that person was just absolutely dedicated. I have to give those folks kudos, because a lot of vegetarians aren't comfortable handling or preparing meat for human consumption. And so, you know, just being aware of who's on your team and what they particularly, what their specifics, what they're comfortable with. Like I said, I'm good with maggots, not so much with human mouths. So everybody has their own quirks and their own preferences. And build your team with the people who really want to do this, and the people who have been trained to do it. So now we're going to talk a little bit about appropriate PPE. You need to be thinking head to toe. Feet. Tyvek booties. Now, a lot of the Tyvek suits come with booties attached. I can tell you from experience, you can walk in those suits with little booties on them for like an hour, maybe, and they'll start to wear through. Truthfully, they're worthless. Truthfully, they're actually designed for like electronics clean rooms. They were never designed, or they were designed for polished floor surgical suites. They were never designed for what we do, which is tromping around outside in sticks, brush, mud, dirt, rocks, ice, and snow. They don't stand up well. I strongly encourage scrubbable boots. I also encourage boot covers. I'm a fan of putting the boot covers over my boots because that keeps all the yuckies out of the tread of my boots and actually makes cleaning a whole lot easier. Instead of having to scrub 15 minutes to get stuff, mud, dirt, blood, chunks out of my boot treads, I take that little plastic cover off, stick it in a garbage bag, and I'm like, yeah, I got a few spots on my boot, and it scrubs off pretty easy. Think about your ankles. If you have different boot covers or boots than the Tyvek, which again, I recommend not using the attached Tyvek booties, be sure that your Tyvek's long enough to cover your ankles. There's, you know, if you look at universities, research labs, a lot of the old school veterinarians are all wearing fabric coveralls. And certainly, when you take our human medical professional, a lot of those guys wear fabric scrubs. Those folks have been trained to do a very specific clothing removal protocol. Many universities actually have laundry facilities on site. A lot of folks that wear coveralls when spraying weeds. Like I know a lot of the county weed departments have a washer dryer on site so that their employees are not taking whatever it is that's on them home. So if I'm going to be traveling and going somewhere, I am going to commonly be using Tyvek because I'm not going to really want to deal as much with highly contaminated clothing and trying to get it home on an airplane or in the back of my truck even. So think about your gloves and think about wrist exposure. And that's a huge one many of us don't think about. We're like, oh, gloves, we're good. Your face, again, you guys know this now with COVID. Think about your mouth, nose, eyes, and ears. What's your splash risk? We talked about splashes a little bit from the hydatid cysts. And again, the risk to people is extremely low. I'm still not going to rub my face in it. Tyvek hoodie, a lot of Tyvek comes with a hoodie. I was teaching a group of folks how to do necropsies. We host necropsy training, several locations around the country and several times a year. The last one I did, of course, was pre-pandemic in February in Colorado. I hosted one a couple years ago, actually, at the National Bison Range. Maybe it was just a year ago, actually. I think it was just a year ago at the National Bison Range. We were doing some elk work there. I do host these around the country, and if you're interested in attending any of those trainings, don't hesitate to send me an email. I'm super happy to let you know when we're back online doing this. That's funny. Back online doing this in person. That made sense. But happy to have you join us. We open those to literally state, federal, whoever would like to come. Public health. Sometimes we have public health veterinarians attend. I've had university students. Anybody who's interested in attending, do feel free to send me an email, and I'll be sure you're on our next invite list as we start to gear back up. Hopefully sometime next fall would be, assuming that the vaccine is effective and everything. I'll go back to the Tyvek hoodie in one of these trainings. I'm walking around the floor kind of talking to people, and there's this guy who's got his hoodie, you know, back over the back of his head, and there's a huge chunk of liver in it. And I'm like, how did you get a chunk of liver hung up in your hoodie, your head? And I don't know if this particular person was doing like the showy Japanese steakhouse thing. I have no idea how a chunk of liver ended up in the little hood behind his head, but there it was. So my recommendation is do not use a hoodie unless you actually need to cover your head. Certainly, anybody working with COVID right now, a lot of folks are wearing hoodies just to try to prevent any exposure to anything or those droplets that they might carry home. But I recommend if you buy Tyvek, don't buy a hoodie unless you think you're really going to use it. It's just really a goober trap in the end. We talked about hair and ponytails. You're just trying to keep your hair back is a pretty good one. The whole flinging your hair through the room is never a good thing.
Lee,
So jump in.
There was a question about disinfectant spray for boots. Do you recommend any disinfectant spray, or do you just recommend scrubbing?
So I have a couple of different recommendations for that, and I actually have a slide for that.
Okay.
In a nutshell, so that's a perfect question. So I tend to use a veterinary disinfectant. There's a couple of them. Now, the one thing you have to be careful of is so for me, because I travel around the country doing this, I'm on the road half time. And so I have to ship stuff like to West Virginia or to Oklahoma or wherever I'm going that following week. And some of the really good disinfectants are considered dangerous goods for shipping priority overnight. And as much as I travel, I'm usually shipping stuff ahead, a few days ahead. Some things you can ship by ground, but not by air. So when you're shopping for the best disinfectant, there's a product called Virkon. If you can get Virkon, it's great. They'll ship it to you. And if you don't have to ship it anywhere else, it's fantastic. It's very broad spectrum. It handles a lot of viruses and bacteria. It's pretty easy to deal with and handle. But it's, again, it's hard for me to use it because if I'm going to West Virginia and that site doesn't have it, then I have to somehow figure out how to get it there with me. We used to have a product called Rocal D that used to be available in all the ranch stores. It has been pulled off the market. They've replaced it with something called D256. I haven't actually tried that yet. I was reading as to what different pathogens. It's not quite as broad spectrum as the old Rocal D was. For chronic wasting disease, and I'm happy to share this if folks want to reach out to me if you haven't already heard about it, there's a product called CIP 100, CIP 100. And that's something that I am now using. It has been documented and confirmed to inactivate chronic wasting disease prions. Bleach will too. A bleach solution will. If I recall correctly, there's a table, it's a publication that has different concentrations of bleach and different amounts of time. And I'm thinking it's like 40% bleach for 10 minutes. If you leave your instruments, if you have stainless steel instruments, if you leave in that bleach longer than that, you're gonna pit them and corrode them. And I've accidentally done that myself. So I switched to CIP 100. It's less corrosive. It's certainly more expensive. It's about $100 a gallon. But I don't feel like it burns my nose hairs as bad. I will tell you though, if you have rubber boots with camouflage paint, when you stick your boot in a boot bath with CIP 100, it will take the paint off. So there's a variety of things. And so really it should depend a little bit on what you're working on. I'm a fan of, I use rubber boots and I put boot covers on top of my boots. I'm also a fan of a boot bath. If I can't do that in the field, I will actually take my boots off, be sure I have like my tennis shoes or hiking boots nearby, and I'll garbage bag. After I take my boot covers off, I'll garbage bag my boots and then scrub them back in the office. So hopefully that answered that question in a bunch of different ways because there's a bunch of different scenarios that you might want to consider.
Thanks, Lee.
Yeah. So we're going to go through a couple of slides here. We're going to talk about the appropriate PPE. It should fit. If it's too short and your ankles are showing, if when you put it on it tears and you feel the need to duct tape it, you really need to probably be getting a different size. And I will let you know that some of these photos are of a team of USDA veterinarians. This one is not. This is another one to think about though. When you're doing work on larger animals, you're often bending over. And seriously, Tyvek was not manufactured for people as tall as I am. I'm a bit of an Amazonian freak. I have to get the quadruple extra large so that it's long enough and does not rip when I bend over. So do be aware and know what size Tyvek you should wear. Even if you don't wear it often, there's times you're going to want to have a little bit of it on hand. So I don't know about any of your programs or if any of your programs require fit testing or health testing for respirators. And we'll talk a little bit about the difference between respirators and masks, especially timely now with COVID. OSHA requires that a, quote, respirator, which has a specified filtration rating, okay? It has to have a certain rating that we are required to be fit tested. Part of that is also educational. If you have a full beard and put an N95 mask on, And I cannot tell you how many pictures I've seen of mortuary workers and hospital workers wearing giant beards and an N95 on top of that. It's not sealing to your face. It's not sealing to your skin. And you might as well not wear a mask at all. The mask is basically protecting you from large blobs of snot and slobber. But you're not getting the N95 protection from the aerosols itself. This particular photo, there's an exhalation valve. I don't know if you've seen in the media now. So this guy wearing this mask is getting protected. If he's infected with COVID, he's exhaling and still shedding and spreading it to other people. In this particular case, if we looked closely, we could see on that stamp that it is an N95 and that it should require a medical evaluation and fit test. Now that said, most organizations or departments are going to have some sort of a way to cover themselves for litigation and still let their employees wear these things. So I have what's called a voluntary use agreement. I just signed something that said, I want to wear the mask. I don't want to get evaluated and fit tested and go through all of that. And I'm willing to take the risk personally. I will not sue my employer if I get infected with something because I wore my mask improperly. That's basically what that's all about. You know, if we were to educate folks, like when they go to the hardware store and buy a mask, and in fact, a fine sand and dust mask that you buy at Home Depot, in fact, that's been some of the ways people are getting N95s. A lot of those actually are rated N95 because the fine particulate sanding masks actually meet that same particulate rating. And early in the pandemic, getting those literally construction worker masks, they are N95s. It's the exact same thing. But if we were to educate folks about, yeah, wearing that with a beard is not very effective. And you really can't assume that it's a perfect seal. And certainly there are some people who have a unique face shape and they're getting so much air moving around the edge of that mask, it's not doing them any good. My husband has a kidney transplant and we did a curbside blood draw this past year. His docs were like, yeah, we really need to get your blood drawn. It's been 6 or 7 months. We need to keep track of your kidney function and the anti-rejection drug levels in your blood. And we weren't going to go in the hospital. It was insane. It still is. And so the people who've been doing his blood draws remembered him and like, hey, I'll just come out of the parking lot. And apparently some transplant centers are actually doing this. My understanding is there's some of them in Missoula, in fact, that are doing parking lot blood draws for high-risk populations. Here in Bozeman, we're not, but our doc arranged for it. So I asked him about N95s for our healthcare workers here. And he said he'd been issued 4 N95s in March, and that's all he'd been given. So, you know, when we talk about them being available in Home Depot, certainly I know that there are people out there that are still going to Home Depot and getting them that are healthcare professionals. It's my understanding that our national stockpile's way back up and that they're more commonly available. I've actually been able to find them online. Within the wildlife community, and I'm talking about this in detail because in the wildlife community, we now have recommendations that we should be wearing N95s if we're handling ferrets, N95s if we're handling mink, if we're doing bobcat research or mountain lion research, that we should be wearing N95s. Certainly bats, any of the bat species. And so it's super important to talk about masks and how they fit well, because if we infect our local bat populations and that becomes a reservoir for COVID, guess what? Guess we're at fault for that. So just something to think about. The rehab groups that are bringing in bats and even feral cats. And there's quite a few rehabbers that work with a lot of mustelids. So just some things to think about, not only in terms of general risk to your own safety and health, but also, especially here in this era of COVID, of not reinfecting or of infecting a wildlife species that does not currently have it. So if we were to look at this particular mask, you know, my employer probably wouldn't tell me I need a medical evaluation and fit test. Some of the secret ways you can tell, if it has 2 straps, it's probably rated, like 2 separate straps as opposed to an ear loop. It's probably rated for a certain particulate, and it's considered a respirator. If it's got just ear loops or a single strap, it's probably what they call a dust mask or a lawn and allergy mask, and it just means lower filtration. So why do you think we might talk about a medical evaluation for masks depending on the level of filtration? Why do you think that matters? I'm asking you to jump in here.
I don't know. I was going to ask you what the medical evaluation really relates to for these masks.
Excellent. Excellent. So you're going to ask me, and I asked you, and I'll tell you, because if you have COPD or if you have asthma and your job is search and rescue, and you're going to be carrying a 250-pound person who broke their ankle at the bottom of the canyon up the mountain wearing an N95, you might suffer some negative effects. For people right now who are saying that they can't breathe with a mask, certainly there's a ton of evidence out there that regular masks and certainly cloth masks don't inhibit respiration very much. Surgical masks, that's why they're not required. And so this particular photo here is a surgical mask. It provides a kind of an intermediate level of filtration, but that's why they put those exhalation valves. That's part of why they put those exhalation valves in some of the N95s. And if you get what's called a P100 mask, you can't get it without an exhalation valve, because if you're doing extremely strenuous activity and you're wearing an N95, it can affect your respiration. It can put a slight extra strain on your heart, especially if you have asthma, COPD, if you've been smoking for 50 years. So there are some health, the medical evaluation is honestly, I think is best used to inform you as to what your risks are. If you're working in 105 degrees in full PPE in a Tyvek suit with safety glasses and an N95, and I've done that, believe it or not. I've been on some Superfund sites where the level of arsenic in the soil was insane. And every step you took, you're releasing this cloud of dust, and you know you're sucking in a ridiculous amount of arsenic. You're darn right I put that mask on. You know, I had my goggles on. I was in the full spacesuit. But it was important for me to know that I did not have lung cancer and put that mask on. So that's the whole point of the medical evaluation, is that you do understand under certain circumstances there is risk to your health. And a lot of employers are going to require that medical evaluation, or they're not going to let you wear it during work hours. And that's how, you know, a lot of employers get away with it, is they say, you sign a voluntary use agreement, you just promise not to sue us if something happens to you and you wear this mask. Does that make sense at all?
Yes, it does. It seems like maybe a person could just go get their own doctor to sign off on that. Does it seem like it's such a big deal to make sure you don't have COPD or lung cancer, maybe? I'm not sure.
A wonderful question. I'd like to answer that by saying that there's a lot of doctors, and I would have personal experience with this, who'll sign just about anything. When my husband had kidney failure, he was told he had the flu, and his blood pressure, by the way, was like 200 over 180. I mean, he was literally in stroke range. And that particular doctor sent him home with blood pressure medicine that would have further damaged his kidneys, and told him he had the flu and to take some aspirin. So we have to remember that there's some MDs out there that got their degree with a D- in school. And so when you're talking about the safety of somebody and an employer approving you to wear it as part of your work duties, they are going to want you to go to an occupational safety and health expert. Those guys have these special sense that they'll put the mask on and test your fit. They can actually teach you about, you know, the facial hair. One of my coworkers right now is actually struggling with the elastic straps on the mask, because even if he has a little bit of a 5 o'clock shadow, I mean, he shaves before he puts a mask on, but by the end of the day, his stubble will actually weaken the elastic. So those are certain things. He might be a better candidate for a different type of a mask with a different type of elastic. So most employers are going to want you to go to an occupational safety and health person. You know, they're everywhere. You basically go and you pay $100 and they spend a couple of hours with you. Sometimes they'll do an EKG, but it's pretty specifically focused on your respiratory volume. They do an inspirometry reading. Some will do an EKG, and then they'll educate you about the mask. Certainly, not everybody is required, and certainly you can go to Home Depot and buy a fine dust sanding mask, and nobody's going to come chase you down with a stethoscope. But I am letting you know about that there are real risks, and some employers will want you to get the medical evaluation, and why. Great question. So what's wrong with this picture?
He's touching his mask.
Yes. If this person was in a room full of super spreading people who've been slinging snot and slobber, he just reached his face out and he's fidgeting with his mask. Absolutely. So when we do wear masks, we should consider the outside of them hot. We're talking about hot zones here in a minute. And certainly for myself, again, with my husband with a transplant, I am the queen of curbside. I don't go into public buildings if I don't have to. I'm pretty careful. But I will think about how close was the outside of this mask to another human being. And so that's something to think about if you're working with wildlife disease, with carcasses. I certainly am not going to be putting the mask on most wildlife disease work. There's a couple of situations where I might. I've had one case where I opened up a carcass and I was like, you know what, this looks frightening like TB. Tuberculosis, I'm in a TB-free state. There shouldn't be tuberculosis here, but you know what? I'm going to keep cutting into the carcass. I'm going to make the decision to continue to do my work, but I'm going to put my mask on. So I've done that once in my career, but it's good to know. And if we get bird flu in here that affects human beings, we know that's a respiratory and aerosol transmitted. So that'll be one. If we get bird flu that's transmissible to humans, and if you end up being a person that is participating on the sampling teams, you're going to want to have a mask. So what do you think the deadliest animal for humans is? And I bet some of you know this.
Primates.
I think other humans ranks pretty high on the list. There's a couple of lists out there.
Monkeys anyway.
Yeah, there's a couple of lists out there, but almost all of them have one particular animal on top. Mosquito. As a vector, it transmits a lot. And that's kind of what a lot of people really went to that research question of, is COVID possibly transmitted by mosquito? So right now it looks like a fabulous no. So just to remind you, PPE includes bug dope. At least long pants. There have been times that I'll put on a Tyvek suit because of the number of ticks. And tularemia is a huge risk. If I've got a tick-laden moose, you darn tootin' I'm going to put that Tyvek on, because now that that moose is dead, those ticks are looking for the next warm thing to walk by, and that's me. And I might even spray my ankles and wrists on that Tyvek with bug dope. If I'm handling dead bunnies, I'm going to be putting on Tyvek, and I'm going to be spraying my legs and my wrists and gloves, everything with bug dope, because I don't want plague-ridden fleas or tularemia-ridden ticks crawling onto me, because once their host is dead, they're looking for the next warm blood meal. So you guys have a protocol. It has some pictures of putting on and taking on PPE. I will say that the most common problem that I see with people with gloves is actually, when they've got gloves on both hands, taking one gloved finger and sliding it under the edge of the other gloved finger. Well, the one gloved finger is hot. So you want to actually use that hand and grab the palm of the glove, of the first glove that you're taking off, pull that hand out, then look at your finger if you had I'm a sci-fi fan, I consider it a breach in the hull. If you've got a breach in the hull and your fingers are yucky, you probably want to ask an assistant, ask somebody for a baby wipe or squirt some Purell on there before you go sliding it up down your wrist to get the other glove off. So the majority of exposure events occurring when you're taking PPE off. And glove removal. So during the Ebola issues, that was the #1 source of exposure for healthcare professionals, was taking PPE off. You think you're, oh my gosh, that was terrible. Whew, glad I'm done with that. You feel like your risk is less, and you forget that the entire outside of your own PPE is hot. You're tired. You might be hungry. You're in a hurry. So I encourage you to take a look at that protocol. So this goes to the wearing PPE appropriately, and we all know what's wrong with this. There's more than one hole to your head. Now, that said, before the COVID pandemic, I had to look really hard for this picture, and now I can find it on popular media every single day. This also goes to wearing a mask, even over your nose if you have a full beard. I love this one. This is somebody who actually attended one of these training classes that we had in person. Shoes, appropriate, sensible shoes. It's a good term for it. So what's wrong with this picture? There's several things. How do you guys jump in on this?
Duct tape.
Yep, we talked about the duct tape earlier.
They're wearing a glove, but it's not a plastic or latex glove.
Exactly. They've got ski gloves on, and I was there. I'm taking the pictures. It was indeed cold, but I really hope they're going to bag and throw those ski gloves away as disposable.
His cotton hoodie sticking out of his Tyvek.
Absolutely. Cotton. I mean, if we're handling something hot enough like this, guys, where we're wearing N95s and eye protection and we've got our hoods up, we shouldn't be exposing our clothing. Absolutely. Good eye.
It looks like he's worn that suit a time or two before. It could just be used from the day, but it looks used and abused.
It's pretty rough. And so take a look at the sleeves. He's got a plastic palpation sleeve and an apron on, a plastic apron on top of all this. But you can see that that's clearly not enough coverage. These are really, really good observations. So they are actually handling live brucellosis. This is an infection, a controlled infection study. So these guys don't know I'm using them in the presentations. But I was like, oh my gosh. And to my knowledge, none of them have been infected. I will tell you though, that I worked in Yellowstone early in my career, studying brucellosis. And we looked at sites where calves, bison calves had been born. We looked at birth sites. We also found placentas, birth material. We occasionally found aborted fetuses. And I worked with a person in another federal agency. We were two of us on one team, but we worked together, different employers. And her employer gave her crap about how many gloves she used. And they basically said, you don't need to be wearing your gloves that much. And that person ended up getting exposed to brucellosis. And I didn't. And I told my employer, I'm like, you know what? If it's bad enough that, I should be wearing gloves. I'm not going to use gloves over and over and over again. I can't tell you how many times I see people try to carefully pull their gloves off so they can reuse it. Don't put it on. If you're going to try to reuse stuff, every time you reuse it, that's increasing and tripling your exposure risk. You're probably better off just to shove your hands in there and then just hose yourself in Purell when you're done. Really, reusing PPE, you feel like you're protected and you're not. Unfortunately, with COVID, with masks right now, we don't have enough masks, so we are reusing them. Healthcare professionals are having to reuse them. We're having to go through some pretty specific protocols to do that. And so always think about that. Remember, the outside of your PPE is hot. And when you take it off, that's your risk. And Lord knows if you try to put it back on, it is. All right, what's wrong with this picture?
Exposed skin.
Yep. And the blue fleece sticking out of, this is a double risk. Not only we expose our own skin, we've exposed our clothing that we can take it home. If the bacteria is bad enough to actually eat through the guts of a bison, you might not want to rub it on your wrists. What about this? See, it saved the worst for after lunch.
Touching his phone with a contaminated.
Yeah, you're gonna get contaminated when you touch your phone after you take your gloves off.
I cannot tell you how many times I've been doing classes and people will be suited up and their phone will ring. And without thinking, zip, they unzip and they go with that bloody glove and they dive in their pocket. I've seen that very commonly. I encourage you, if you're actually going to be working with carcasses or working with necropsies or collecting carcasses in the field, leave your phone in the vehicle or assign somebody, and we'll talk about that in a minute. If we have teams of more than one person, we can have someone who's a clean person or a cleaner person manage that phone for you. You guys ever heard of phantom pocket vibration syndrome? It's real. No, it's real. It is hilarious. But there are apparently people, especially with social media, where your posts are liked, and especially with you know, texting. A lot of people will be having text conversations with 6, 7, 8 other people at a time. I don't have an attention span that long, but you know, there are folks who really do that pocket vibration is a sensory stimuli, and there's actually been some studies that suggest that a lot of people's mental health is now dependent on how many likes they might get in a day, that there's a generation of folks coming up like that. And so again, if you're going to be working in this, just hand the phone to somebody else and they can tell you, oh, you've got 3 more likes, and that makes you feel just as good. You just don't have to handle a phone with your bloody gloves. What's the moral of the story here?
Clearly weren't using the proper PPE.
They actually, it's a great point. They were using the proper PPE, but they were not using their PPE appropriately. Tyvek's not waterproof. If you apply pressure to it and kneel in a pool of blood, it's gonna soak through. So to your comment, not using the proper PPE. I have 2 people that I work with on a regular basis that have back problems or knee problems, and they can't actually work on large carcass necropsies without kneeling. And so they did get appropriate PPE by buying foam, disinfectable, dunkable knee pads. If this duck were infected with a zoonotic strain of baby influenza, this person would be in trouble. So there are people who tend to you know, really want to spend more time with the animals. I have one person who, when they do necropsies, just cannot resist opening the uterus, pulling out the fetus, and rocking it. So those folks, you just need to talk to them a little bit and be sure they understand the risks, and that you may need to give them a little bit extra PPE, and just understand the complexity of your team and the diversity of the team and their experiences and their perspectives, and help them understand the risks. And if they need additional PPE, be sure they get it. So here's a question for you. Do I have to fully suit up every time? And the answer is that we need to assess the risk. So in this particular situation, is this person using a proper PPE?
What is on her nose?
Oh, that's a wonderful question. Actually, there's nothing on her nose. I have never had anybody ask that.
Oh, It's behind her.
Yeah, that's a pin flag behind her. Oh my gosh, I've just never even noticed that. I might actually have another picture of that.
I thought it was a clip or something.
That is hilarious. So nothing on her nose. There's a pin flag behind it. Wow, I wonder if I can like Photoshop that out or something. Looking at what she's doing. Is she doing it right?
No,
What's she doing wrong?
needs some sort of face covering from the smell, maybe coverings on her shoes.
So the covering from the smell, I mean, this is, and I'm just letting you guys all know this is a trick question. So if this were from an animal that we thought were infectious or had demonstrated clinical signs of disease, we would absolutely be considering face protection. We'd be considering aerosols. We could be considering splash protection, and we might be protecting our clothing with Tyvek. If we saw that this was from the term is an apparently healthy animal. So that's a trick question, and you answered it correctly. But now the question becomes, if this is from an apparently healthy animal, is this person doing the right thing?
Yes.
Pretty darn close. Pretty darn close. This person may be actually collecting insects off of the top of a bison pie. This person's actually collecting fecal samples from a bison pie. But there are people who run around and collect insects off the top of bison pies to study them. I would still have recommended boot covers because they're easy and they're not difficult to deal with. They're not terribly expensive. And you know, the chance of you stepping right in that pie as you're sampling, it's extremely low. Her body posture, she's got her gear behind her. She's got a person standing far enough away in case there's an accidental flinging of poo. So she's got it set up just right. If I were in her shoes, literally, I would think about boot covers. But then you also have to think about how far did she have to hike to get to this spot? She might have had to hike and stepping around and through 50 bison pies to get there. And you're not going to hike in boot covers. They're not going to last. You would get, you know, it wouldn't be effective. It would, in fact, add risk. It'd be slippery if you're crossing a stream in boot covers. So that would be the one that I would think about, and it would certainly be situationally dependent. So in this particular situation, she's done a pretty darn good job. This is a healthy animal. She has pretty good posture and position. Consider boot covers, but under the circumstances, if this person hiked 10 miles to get there, I'd still probably put boot covers on, bag them, and pin in my bag, just because that way I know I haven't accidentally spread something out of that location. But under this particular situation, we're probably in pretty good shape.
Lee, we have a quick question. Do you essentially buy boot covers from the same vendors that supply Tyvek suits?
You can, and I actually have a list of vendors for stuff with some links, and I don't know if I sent that to you. I'd be happy to send that to you when we're done. Now, you know, of course, the federal government cannot in any way recommend any vendors or any companies. So my list includes 3 or 4. I will tell you that some of them will give you a pretty good discount if you call and ask. You can get a bulk discount. And so I actually have different suppliers that I use for different things, but I honestly shop around quite frequently. And so my vendor list gets updated 3 or 4 times a year.
Okay.
And again, it has multiple options and links. What I do provide though is some tips. So there are some boot covers out there that will only fit people if you have a shoe size of a toddler. I mean, they're just not made for adults, and they're sold as adult boot covers. So I don't include those on my list. And the Tyvek, you know, there's different sizes. Now, the other thing with Tyvek is there's different levels of splash resistance. There's super light Tyvek that is really more of a dust preventive for people working with electronics. And then there's like A20s, which are fairly splash resistant, have a little bit of that plastic Tyvek-y feel. There's A40s that are even more resistant, but again, then you can kind of get hot in those. I'll say if it's 20 below and it's blizzarding, I'm like, give me an extra layer of Tyvek, you're loving it. But if you're in Oklahoma at 105 or Kansas at 105, you just going to think about those things. So I can send you a spreadsheet that has those links and that information. Happy to. If I don't think I included it, but I'm happy to do that.
Okay, yeah, that would be great. And then there is one more question relative to collection. And the question is, if collecting scat samples for DNA or other dietary analysis, would you treat each sample as from a potentially sick animal? And then what PPE would you recommend?
So anytime, it's a wonderful question. Anytime I collect scat, I'm going to be wearing gloves. And so when we define sick, certainly we know that a lot of wildlife species normally have parasites. A lot of wildlife species have tapeworms, that's part of being a wild animal. Wild animals are not sterile out there in the landscape. Is that animal clinically sick? Is it shedding an organism? And is the organism potentially infectious to humans? Relatively low probability. But it's not hard to put on a pair of gloves. So I would absolutely, if I'm collecting scat for DNA, and I have done an awful lot of picking up and packing out grizzly bear poop, and there's a bear scat's just nasty anyway. I mean, anybody, well, clover scats aren't bad, but in general, bear scats are pretty nasty, and you're going to want to wear gloves anyway. But again, that's where I'm probably going to be gloves and boot covers with most of it. And then just be careful to not fling poop. Don't throw it at people. Don't eat the wolf poop. All of those, the basic precautions. If you're super shaky, the other thing I'll give you a tip on is the size of the opening of your bag or your container. I've been doing some necropsy training where some folks showed up and they had bags that literally had, you know, an opening the size of a quarter. And to try to get a sample into a teeny tiny itty bitty bag like that, you're going to hose the outside of the bag. You're probably going to hose the person holding the bag. It's going to take more time. So I encourage you, if you're taking samples, get a big bag. It just makes it easier and safer. The other thing is with shredder bags or medium-sized garbage bags. Those can be super handy as an extra layer. And you can actually, if you forget your gloves or if it's 20 below and some of your gloves happen to have cracked, you can always use a bag as a glove over your arm. So yeah, I'm a gloves and boot covers kind of person as a bare minimum. And part of it makes me sleep at night knowing that I'm not the person that let that disease get transferred to another piece of land or to another population of wildlife.
Thanks, Lee.
Sure. So we're talking a little bit about the tools, instruments, containers, disinfectants. Most of us can't carry buckets and brushes out in the field. Which is where I mentioned if you can use those boot covers, dispose of them. And if your boots are yucky, for sure, I just bag them. And then I always have a separate pair of shoes. And we'll talk about our zones here in a minute. But don't skimp. That's that whole reusing gloves. Just don't skimp on it. If you're going to do it right and be safe. So let's talk a little bit about zones. So if you have a carcass, you're going to consider that a hot zone. Okay. And then you're going to have a warm zone. Then you're going to have a cool zone. You're going to show up in your truck and you're going to park in the cool zone or outside. Now, these are all clearly, it's like progressive lenses as opposed to bifocals or trifocals. The boundaries are actually really a little blurry, but that's part of your job is to try to define them for your crew as best as you can. I tend to use my vehicle. Anything beyond my vehicle is as safe as it's going to get, and I'm hoping it's as safe as my home is. The blue zone should be as safe as my home is, hopefully. Yellow zone, yeah, we're kind of passing some stuff back and forth, maybe some instruments. Maybe I've got somebody taking notes there. Maybe that's where my sample kit is. And so we're going to set up, and we're going to put on our PPE. And we're going to put on our PPE as we enter into the warm zone. Ideally, our PPE is on the edge of the cool zone. And if you're working with folks who have back trouble or knee problem, I encourage you to have a chair or the tailgate of a truck to help people put their PPE on. You're going to move into the hot zone. You're going to do your work. You're going to come out. You're going to take your PPE off, dispose of it. You're going to decon, disinfect your stuff, get in your truck, and drive home. So ideally, it's you. And if you're really lucky, you've got a helper. And that helper is going to maybe, if it's a large animal, maybe actually help you in there on the carcass. If you're super lucky, you have a third person. If you don't, send your helper into the warm zone because they can hand you clean instruments. They're going to have gloves on because they're probably going to get something on them as you move things back and forth from the red to yellow zone. They may be holding your sample baggies and you put the sample in it. Oops, sorry, I just got some blood on your glove. Or I just got some maggots on your hand as I put the dead duck in the bag. But having that person in the warm zone, they are cleaner than the person in the hot zone. The problem is a lot of us might have people, perhaps media, or a reporter, or someone who just thinks they're generally important, and you might ask them to stay in the cool zone. Do not trust that they will stay in the cool zone. Know your people. If you've got some sort of a film crew or media crew that you're not familiar with, or a group of students that you're not familiar with, if you're not sure, suit everybody up. Because I can't tell you how many times we've had folks, oh yeah, I'll stay out here. And if I'm working in a shop, I'll actually try to block a boundary between the hot zone and the warm zone with kits, boxes, coolers, action packers, whatever I can to try to control the traffic, try to control the flow, and make it more difficult for a person to get into the hot zone with me, and that I can see them coming into the hot zone with me, because I want to control that. So you got to be prepared to manage other people. And if you're not sure of what you're dealing with, suit them all up. So risk assessment and expected level of exposure, because we don't know what diseases we're dealing with. And we ask that question, is scat? What's the risk of scat? And there is a risk, but we're not sure what it is. What we do need to do is assume the worst and be prepared we need to have first aid with us. That's why I always recommend at a minimum boot covers and gloves, because even if there's something that's not particularly infectious for humans in terms of a systemic, this is going to put you in the hospital. If you even cut your hand handling scat or handling a carcass, there are plenty of bacteria that will give you a very unpleasant and nasty local infection. I had one person who tended to not be as vigilant in checking their gloves for nicks and cuts. And they ended up doing about a week's worth of necropsy work. And I got a call and they said, I've got all these weird, like, kind of pustule blisters on my hands. And I'm like, oh my gosh, how careful were you with your gloves? And well, I'm sure I put one pair on and I wear them all the time, but when I take them off, there's usually blood on my hands, on my skin. I'm like, that is not good. We need to talk about checking for breaches in the hull, because you probably have nicked yourself several times with that knife, and it's probably not a terrible fatal infection, but you probably ought to go to the doctor. They may want to lance wash and clean those wounds, and they might even give you a round of antibiotics, depending on how much of this you've got on there, because you have probably injected yourself with a knife tip with some bacteria that's trying to invade your body. It's not good. You're probably not going to be really severely ill, but really check those gloves. Assume the worst, wear those gloves, check them, be prepared, have some first aid with you. I carry those antibiotic Band-Aids with me. I consider that my first line of defense. If I accidentally nick myself, I stop my work, I go out into the warm zone, head to the edge of the cool zone, I take off those gloves, I immediately use a baby wipe, I'll put Purell on there to try to disinfect the wound, and I put an antibiotic Band-Aid on it. So just even having those on hand in your vehicle or in your kit is super handy. I tend to use a fishing tackle box. It's pretty easy. You can shove it in a backpack. I can usually do, 8 or 10 necropsies out of it if I need to, and I can hike with it. So just having some of those Band-Aids with you is super handy. And plan for safe disinfection and disposal. We talked about garbage bags and how handy they can be if you need to have an extra glove. Say your gloves all blew out of the helicopter. If you've got a garbage bag, you always bring extra. If you're not going to use them for disposing your stuff, you may end up using them for an extra set of gloves or extra bagging of samples. So the Wildlife Health Center website, they have this little card that you can put in your wallet. I know a few folks who've taken a photo of it and have it on their home screen on their phone if they're doing necropsies commonly or working with plague commonly. I do carry this with me, and I've actually personally used it in the emergency room twice now. I was a little worried because I thought they'd be like, what do you mean? You can't tell me what to consider. Oh my gosh, they were so grateful. They're like, are you serious? Holy cow. So we're 5 years out now. I had a pretty severe event of vertigo. Was driving, couldn't see 2 feet in front of the truck, had to pull over, laid over on the side of the road, literally got out of the vehicle, slid down to the ground and laid there for a while. And eventually, after about 10 minutes, went away. I had no cell signal, drove to a spot with cell signal, called my husband, went to the ER. And I had actually suffered a tick bite in an eastern state not too long before, so I had a pretty good welt. I'd done some necropsies on some brucellosis infected animals. So I handed this card to them and they were so grateful, like, holy crap, we can run like 5 tests right here, right now and rule those out. In the end, it turns out I have a congenital hole in my heart. I have an atrial septal defect that I've had my entire life and didn't discover until I was nearly 50. And that's what caused that. I actually had a blood clot go backwards. But the ER folks were tickled to death. So in the ER, they're putting pins in my toes and I can't feel it. I'm watching them stick a needle in my toes and they're like, oh shit. They were so grateful for this list because they're like, I can run down this list real fast. And if we've got something that's on this list, we'll know what to do. So I encourage you to think about it. I will let you know that I have had nothing but gratitude from healthcare professionals when I've given them this card, when I've done that twice. So you guys have these fact sheets. We can talk about disinfectants. I'll send you the vendor list. I think I've listed them all off here. Use the disinfectant appropriately. There's a difference between cleaning and disinfecting. Disinfectants will not penetrate large globs of dirt, mud, blood, chunks, poop. So you got to scrub all the stuff off before the disinfectant will work. Cleaning your instruments is a huge risk because that's where you're likely to cut yourself. And you're also really tired. If you're using boats, think about, and this is not only obviously for our aquatic invasive species, but for diseases like cholera. Think about getting a hand sprayer, a little pump sprayer, and spritzing the outside of that boat when you load it up on the trailer. Same thing for sample containers. Just wipe the outside. I'm a fan of baby wipes, Purell wipes. Certainly baby wipes have an antibacterial in them. They're not great as an antiviral, but the action of just wiping the outside of the container does a whole heck of a lot. There are Purell wipes that are completely antiviral and antibacterial. They're alcohol-based. They do evaporate a little bit quicker. The other Purell wipes are a pretty good disinfectant, but they don't hit all the viruses. Again, the whole process, it's not ever going to be perfect. It's risk management. It's reducing the risk of carrying it off-site. And then disposing carcasses, there's a lot of options: incineration, burial. And if you feel that the animal does not have a lot of signs of illness or disease, and you discovered, oh boy, that was likely a shot trauma or was a gore wound, or we're pretty confident it was actually hit by a car, you might decide to leave it on the landscape. Certainly you don't want to leave it near roads because then you encourage other scavengers to get hit. But there are a lot of places that'll pull a carcass off. Those of you who are in grizzly country, certainly there's going to be a lot of different recommendations for that in bear country. So do think about that. But we do know that there's a lot of scavengers that depend on the availability of carcasses. So just make those decisions carefully relative to your disease risk, whether or not you're working with apparently healthy animals or you're doing a disease investigation. If you're doing a disease investigation, I strongly recommend at least burial. And remember that PPE removal is your most dangerous risk, and then thinking about disposable versus having to take your stuff home and wash it. So before you walk out of an area, before you're done, do your final wash up. Soap and water's good. If you don't have Purell, if you don't have wipes handy, soap and water is way better than nothing. You're actually washing majority of the material away. We talked a little bit about baby wipes and hand sanitizer. Do a final site check. I'm sure I've left a few pair of forceps and scalpels out in the landscape. I really do my absolute best not to. I try to know I brought 4 in, I got 4 going out. So think about that as you do that. In general, for your safety, if it's small enough to put in a box, ship it whole. That's for your safety. And it's also for the lab, gets way better samples. If it's all in one package, the lab can do the necropsy themselves and examine all the information and not depend on your descriptions or photos. And you should have a shipping sample packaging and shipping protocol there. And that is based on not only what the lab's like, but also what's the International Air Transport Association's requirements. So like FedEx and UPS by air. If it's too big for a box, and if it's not something that you're concerned about as a zoonotic risk, like a completely tick-covered moose and tularemia, you would not want to be opening that one. Then you want to consider getting necropsy training and learning how to do that. And as I mentioned, we do offer that periodically, and just let me know if you would like to be notified on future classes and availability and where they're being held. So we're going to super fast go over outside of containers. And I'm just going to show you a few pictures here. They're kind of like, yeah, bad things. These are what's arrived in a lab. And so you really want to clean them up and you really want to disinfect the outsides of them. Some sites have small incinerators, so you can certainly consider burying on site incineration. That picture on the lower right is from the Mad Cow era in the UK. And they certainly have special incinerators that are designed for that. And some of those special incinerators also may be considered by some state agencies for use with chronic wasting disease. At a bare minimum, if you're going to be using a landfill, you want to disinfect, double bag, and disinfect the outside of that bag again. Again, a hand sprayer is super handy for that. Composting was something that was more commonly done during the last avian influenza outbreak. USDA was looking for some specific composting sites. Done properly, it can work. Just throwing a bunch of carcasses in a pile is not composting. And as we talked, if you're pretty confident you're dealing with a healthy animal, you're not working on a disease investigation, and you had no signs, you may choose to leave the carcass out on the landscape. So for your clothing, again, we talked about the disposable stuff is nice because you can bag it. I do not recommend using clear plastic bags and just throwing in your local dumpster because lots of people will see that and be concerned. I do recommend using a black plastic bag. There are special water-soluble bags available, and that's what this picture is of, and I have some of those. And you can actually tie your clothes up, and I use this frequently when I'm flying and doing training classes. And even if I don't think I have gotten anything on my clothes, if my Tyvek looked good, I don't have anything that's soaked through, I will still actually put my clothes in these water-soluble bags, tie a knot on it, and then the entire bag goes in the washer. And that's pretty nice. It dissolves. It's made of cornstarch, but it keeps the rest of the clothes in your suitcase pretty clean if you need to. It's also, if you need to throw it in the back of the truck, a good thing to do. Boots, again, I recommend scrub brush and dunk it. If you can't do that, bag it. And that's where those boot covers really help making the boot cleanup easier. So just to kind of remind you that, again, everything's connected. We've been over that. We've talked about it. We're living it right now. We are totally living this with COVID right now. And you look at half the diseases we talked about, most of them. CWD has an environmental contaminant component. There's certainly concern with the risk to humans. We have significant impacts to our wildlife health populations, bird flu, all of these. Everything is connected, and the world gets smaller every day. And as wildlife health professionals, we're kind of that first line of defense. We really are, especially with things like avian influenza. If we're discovering that early on before it hits the poultry sector, we have a chance to reduce our impact on our agriculture. So we're really a super important part of that animal health surveillance system, and truly it's an environmental health surveillance system. So I will not spend much time on this. I did want to let you guys give you just a few pictures. And this is when USDA, this was a foreign animal disease outbreak. And I don't know if any of you have ever been involved in a foreign animal disease investigation where USDA comes in and quarantines and shuts everything down. And I've been involved in two, one directly. This was one I was not directly involved with and on site. But you may have heard about screwworm. It was here in the United States in the 50s, and then it came back and nobody knew. Nobody learned it. All these veterinarians, nobody taught them in school what to look for. And it turns out the local vet clinics have been seeing some cases for months before it got actually discovered as an, oh crap, we've got screwworm in our wildlife. And it's in key deer. So the maggots actually eat living tissue. Most maggots eat dead tissue. And this is the last slide, and we're way past lunch, hopefully not thinking about dinner yet. But these particular maggots eat healthy tissue, which makes them really different and kind of scary. And so we have that whole cycle where you have the fly maggot cycle, but the wounds get worse and worse and worse on the animal. And this is what it looks like. This animal actually was alive until it was euthanized. It's pretty horrible. And it's fortunate that we caught it and we got it in time to keep it just in the Florida Keys. USDA set up a quarantine, set up checkpoints. They were very concerned about domestic animals, and the homeless human population was a huge concern. We were so lucky that we, as an animal health community, figured out what was going on before a lot of the homeless people in Florida were infected with this. So just something to think about. I know some of you guys on this call are wardens or conservation officers and may be involved in some of this. So if you end up in a foreign animal disease investigation and USDA is making some decisions, your state animal health folks will be making decisions all the way down to a local level. It's going to be a huge, chaotic, messy decision tree. But you end up doing everything from responding to phone calls, engaging the public, limiting illegal feeding. These particular key deer, there's a lot of people that feed them human food. They are pretty much garbage addicted now. And there's also a large feral cat population in Florida. So key deer have actually learned to eat canned cat food as well. So there was a multi-pronged approach on how to limit illegal feeding, because if you get all of these deer that are covered in maggots, all trying to eat out of the same can of cat food, they're going to be spreading the maggots. The maggots and the flies will infect the next animal. So obviously euthanizing, we've talked about this, the carcass handling and removal. If USDA comes in with foreign animal disease issues, there's going to be incident command stuff, all these structures, all these, chains of command. I personally have not yet taken incident command training, have been asked to. It's something that I know the folks that were involved in this tell me there's no way they could have been effective without it. Crowd control, dealing with public meetings, and there's a lot of work to get out to outlying islands because the water is pretty shallow and key deer are really good swimmers. So just to kind of give you a wrap up, think big, think outside the boundary, think about what could happen and what teeny tiny little steps like putting a pair of gloves on and boot covers can do. Just paying attention. And if you're like on the fence about, well, so I collect a carcass, if you can, go ahead and do it. And worst case, a lab says we can't handle it right now, or you can find a lab that does. And don't ever hesitate to call me, because I work with a variety of labs around the country and I can at least give you a referral. Oh, well, this lab's usually really good about handling it. So, there's a lot of options there and a lot of folks that can help you with that. And dealing with multiple species at multiple scales and really doing that investigation, that field investigation, figuring out the epidemiology is how we figure out if we can do something about it and what we can do about it. And most times we can. Most times there's management steps we can take that really mitigate what's going on and may even in some cases be able to completely eradicate disease. There are some cases of that where we have eradicated disease. Be prepared for the biosafety and biosecurity threats. Think smart about containing it. And vigilance is kind of key. Just keeping your brain on, keeping your head wrapped around the if-then scenarios, what-if scenarios. And it's not to scare you, but it's just to make you sharper and smarter to make better decisions about it. And with that, guys, I want to say thank you again for all the fantastic questions. You guys have really been one of the most engaged groups that I've given this presentation to in the past couple of years. It's super exciting.
Thank you, Lee. Appreciate it. I don't know if you can, well, we can leave that up for a little bit, but I think most folks now have your email address, and I think your phone number is on the bottom of a lot of those documents you sent to us. So definitely appreciate that, and I would guess that a lot of people will get, in touch with you. Thank you all for participating today. It was great feedback and interaction. I'll just put this out there. If you have any further questions for Lee, you've got her information. And you can also reach out to me as well for other things or other resources. So please do that. You guys have my information as well. I want to thank Karen Lynch for helping out with this, getting this all set up and worked the announcement through the website. And with that said, Lee and I have talked about this before, and I hope that we can continue to have this training in the future for those people that could not participate today. I think we'll still limit it to a smaller group so we can kind of manage that participation, but it's been great. I think Karen's on the line here. Karen, do you have any follow-up here?
No, not really. Just glad to know that it was really a very informative session, and I thank everyone for coming, and for you, Robert, also for your good work in setting it up. Thank you.
Thanks so much, Lee.
Absolutely. And again, happy to answer questions. And I also will let you guys know, there've been a few cases where I've had calls and people are like, I've got 10 dead things out here, and I give me a heads up. And so I've been able to say, grab your gloves, grab this, you might grab that, grab a camera. I have some checklists that might be helpful. And certainly I can help facilitate contact to labs. And I will get that vendors list to you. I apologize for not including that in the first email attachment, but I will get that to you. And again, we're not recommending any particular vendors. There's a variety of options there, but hopefully it's got enough information there to help you figure out what we've learned is a more useful model or style versus less useful.
Well, we appreciate that for sure. And I know that there are different capacities on this webinar today as far as employees. So we have biologists, we have law enforcement as well. But keep in mind, when all you guys in LE, just keep in mind that once we're in a situation where you suspect that there might be illegal activity, just make sure that you follow all the recommended procedures that Lee was talking about as far as protecting yourself, protecting the environment, etc. But also remember that you need to follow the protocols when you're collecting evidence, because at some point that could turn into a situation where you've got to provide that through chain of custody to a court. So again, it's just looking at it, at a larger level and what you feel you might be dealing with. So that's all I'll say in closing, just from a law enforcement perspective.
Do we have time for any questions?
Yeah, go ahead, shoot away, guys.
I have two questions, but I'm going to start with this one, and I had to multitask for a couple minutes, so I wasn't always on. But Lee, you had a picture of an osprey in the beginning of your presentation, and the ends of its wings tips were gone, and I was wondering what that was.
Power line strike.
Oh, okay. So they were burnt off?
Great question. Yep.
Okay.
Great question.
And then second, I wondered if there were any handouts created or anything that we can share with the public about potential wildlife diseases, something that's consumable for the general community, just to have that kind of information out there on what are things that people should be looking for. So
like all the things that I provided to you, we've provided as training materials. the things that are available for the public, you're going to really want to look at your like CDC. You're going to have a state Department of Public Health. And you may choose to work with a state wildlife health agency. So like if it were Montana, it'd be Fish, Life and Parks, a DNR. And those guys have, a lot of those agencies have public outreach and or humans dimensions groups that's their job is messaging. And they have done a lot of work with figuring out what works and what doesn't. We know that with diseases like brucellosis and chronic wasting disease, like CBD, you know, how do we message don't transport carcasses and neurologic material across state lines? And so there's been a lot of learning on that in terms of what works. Obviously, recently, our messaging about the use of masks has only been roughly 50% effective in the United States. So I do really encourage you to work with some of those groups. A lot of the things that I've put together, if you look at, I've got references from CDC, like on our harmful algal blooms. I'm pulling stuff off of EPA. EPA, a lot of the federal agencies also, we're all of our photos are basically publicly available, other than law enforcement specific cases, obviously. But you know, those materials and things are posted on sites if they have them. We do not, within the Wildlife Health Office, have a big, giant public-facing website. We don't have the manpower to put one together, and nobody's offered to put one together for us, to be honest with us. There are 3 of us in this program that cover the entire country. 2 of us actually cover the country, and then I've got a biotech that helps me. So we're a really teeny tiny program. But a lot of the state and federal agencies are out there have massive programs that they'll have a lot of that stuff available. And honestly, they've been super fantastic. Like, yeah, here's my templates. Just change it and fix it. And that's part of why I've offered to share a lot of what we have with anybody else. If we all share our templates and information with each other, we'll get the word out.
Great, thank you.
So we had 2 pending questions. Daryl, you want to go ahead and ask yours?
Yeah, I had a question about the chickens you mentioned about it could be a costly mistake if I think the subject was on bird flu. I'm not sure, but I just had a question like what would you recommend for us, like if we were to privately raise our chickens, and what's the safe number to do so?
That's a fantastic question, because backyard poultry is absolutely becoming more common, and even in urban areas. So we have what we consider a domestic animal precautions. And this is especially important if you're living in areas that there may be wild birds. So we strongly encourage folks to reduce contact between their domestic birds and wild birds. And so whether that's just a large fenced pen, I know a lot of folks that have indoor-outdoor coop management, that's super important. The number of animals that you have is a lot less important than the management of waste, litter, those types of things, and what you decide to do with that. If you're going to be doing composting, you probably don't want to do that in an area where there's a high waterfowl population. And then also making sure that when you're tending your backyard poultry, that you're not wearing the same clothes and shoes and boots as you would take to work to use on a waterfowl survey. That's another advantage of those boot covers. So you can have shoes dedicated at home, or muck boots that you're going to use at home, and then don't use those at work. And that's another advantage of the boot covers, is that the boot covers, when you're at work, that's an extra layer to keep you from bringing anything back to the office. So think of your house, the office, your work vehicle, and then the field site that there might be a mortality or disease outbreak. If you actually think about those are kind of your hot, warm, and cold zones as well. So we have that zoning in terms of physical right around a carcass or an outbreak, but there's also, it extends all the way out to your house. And that is an impact to your poultry husbandry at home. Same thing with dogs. and vaccinate your pets if you can. Deworm your pets. A lot of those parasites don't jump and cross into wildlife species, but some do.
Oh, okay, good. That's it. That answers my question. Thank you.
Awesome.
And then we have another question here from you for you from Errol. Errol Standing. You there, Errol? Let me see. I've got his question right here. His question is baiting deer a risk for the spread of CWD?
Definitely. And there's just no doubt.
Okay.
So that's, so I'll follow that up. Are there states that are intentionally baiting deer to try to use that to manage CWD? Yes. So any feeding or baiting of wildlife that increases the density of the animals, definitely a risk. And so when we think about the density of animals, we have to think about the number of deer on a bait station and the swap of snot and slobber, poop and pee, because we know that CWD occurs in urine and feces as well. And then we also have to think about the contamination of that poop and pee, because we get prions in the soil. And there's a really neat publication out there that looks at certain areas around mineral licks, certain areas around historic areas that were used for baiting, and they found higher levels of CWD prion in the soil. I think that work was done in Wisconsin a couple of years ago. If you haven't seen it, send me an email and I'll dig it up and send it to you. But basically, we may at some point be able to literally detect prion puddles in the environment, and not liquid puddles, but areas on the landscape that are hot spots for prions. And right now, based on the preliminary research, even salt blocks are likely to be that, and so are bait and corn stations. There are some states that have justified using that to make targeted culling easier. For example, I believe the state of Arkansas decided to allow baiting specifically for CWD management activities. We know from the state of Illinois was the first state to really make some headway on how to manage CWD. We all know about how Wisconsin was one of the first states to step up to try to manage CWD. And unfortunately, you know, nobody knew any better. They did the best they could. They worked mostly with hunters. They did the best they could. They encouraged hunters to take as many deer as they could. They implemented the Earn a Buck program, and hunters got tired of it. And hunters felt terribly overregulated by the time it was done. And it ended up that, I mean, the first year goal was to take, I think, like 2 million whitetail deer. And I think their first year they only ended up getting a couple 100,000, maybe 300,000. And so it just didn't end up being as effective as they had hoped. And so what most states are seeing now is that working with hunters through regular harvest and to modify those harvest targets really helps. And that includes a lot of the age structure with CWD. Illinois, their state has put out some publications. It's been pretty neat. They've actually been able to keep prevalence, the percent positive in CWD in their deer, down in that 6% and less in most of their herds. If you look directly across the state line in Wisconsin, they've got areas there, hunt districts, that they're looking anywhere from 25 to 50% positive. So we're at a point now with this, we've had this disease long enough that we know that management makes a difference. And we know that concentrating deer on salt licks and bait and in deer yards makes a difference. And unfortunately, our national elk refuge and our feed grounds in Wyoming may become some of those prion hotspots and prion puddles. We know we have CWD in the system. We have not been, you know, detecting impacts to elk on the feed ground elk, but we know we've got CWD in deer. We know we've got CWD in moose not that far south. It's a matter of time. So yeah, I have to answer honestly. A lot of people aren't going to like that answer, but that's the truth. Yeah, it's pretty significant risk, driver.
Wow. Thank you for that. Does anybody else have any other questions?
I'd like to ask a question, please.
Go ahead, Mike.
Hello, my name is Michael. I'm with the Kiowa tribe in Southwest Oklahoma. Our southern border of our jurisdiction is the Red River, the border between Texas and Oklahoma. The EPA has informed us that they want to do a mass culling of wild pigs because they become a nuisance. And they informed us that 50% of these pigs have a brain disease that makes them inedible. And they want to do just a mass culling, but they want to come onto individual Kiowa federal lands to do some of it too and notify the landowners. How can we assure that the landowners can we assure the landowners that they're going to safely dispose of these animals? And how do they do that, the culling of the mass animals safely?
So that's a good question. So is it USDA that's coming in?
Well, the EPA informed us that it might be EPA and USDA together.
EPA is usually not involved in much wildlife work. USDA certainly has a branch that's dedicated to doing that. What I recommend that you do is talk to USDA about carcass disposal and find out what their plan is. Incineration is a good option. There are also an increasing number of what they call tissue digesters. And tissue digestion is something that also will even get rid of chronic wasting disease prions. There are getting to be more and more universities that have those available. I am not sure what disease they're specifically referring to. There have been a couple of diseases identified in feral swine that have some pretty significant risk to people. But I'd specifically be asking about their carcass disposal methods. And I'd be asking them to document if they have specific concerns for a disease that's a risk to humans, that they provide you with information as to how their carcass disposal methods will limit that risk.
I understand. Thank you.
Anybody else with questions? I don't think we have any more, Lee. Again, I can't thank you enough for putting on this presentation for us. It was extremely beneficial for all of us. And we truly appreciate that. Look forward to working with you in the future.
Absolutely happy to. We're just happy to share information that helps keep any of us safe, helps keep our families safe, helps keep our food supply safe. And, you know, I actually went to school in wildlife disease and physiology, and this is not commonly taught. A lot of us have been through school, specifically for wildlife disease or parasitology, and these types of thoughts and these types of decisions and the risks are not well taught, and happy to spread the word. And again, I'll follow up with that spreadsheet. And if anybody else has any additional questions, feel free to email me.
Great. Thank you so much.
Thank you.
Thanks, everyone. Appreciate everybody participating, and you guys all take care and be safe.
Thank you.
Thank you.
You bet. Thanks a lot. Take care.